Hello, 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 everybody. Welcome to stream. Welcome to stream, everybody. Today we have a very special live stream for you all today. We are going to be talking about this concept of genetic novelty, and I am very, very lucky and happy to have on a very special guest. Special guest, please introduce yourself.
Hello, everyone. My name is Dr. Cara Weisman. You should feel free to call me Cara. I am the author of the journal club paper that we will be talking about today. You want me to give a little bio?
Yeah, sure. Go ahead.
Okay, so I did my PhD technically in biophysics at Harvard. At Harvard, biophysics just means any biology that has any vaguely quantitative element. So really, I'm an evolutionary genomicist, and that's where I wrote the paper that we will be talking about. And now I am a fellow, which is a sort of like independent postdoc at Princeton.
Yeah, so one could say that you are pretty qualified given the the pedigree of your education. One could. That's awesome. And I wanted to share a little bit about the kind of the story of how we came into contact. There was this comment that someone left, and I'll just read it out loud. And it says, and this was on my recent video about the birth of new genes, something that I've been thinking a lot about recently. And this comment says, I've worked with someone who is in this minority saying that we haven't found good enough for any de novo genes. proteins. She's got a really interesting paper in which she shows that sequences for many proteins evolve fast enough that information theory limit prevents us from finding gene homologs, even when there is an evolutionary relationship. Her paper, Many but Not All Lineage-Specific Genes Can Be Explained by Homology Detection Failure, is a fascinating read. And this is also evidence that I do read all of your comments. And if you do have scientists that you think would be an excellent interview on this stream, please go ahead and share those with me. So, first slide here. This is my abstraction of your idea, and you can kind of judge me based on the composition of the thumbnail, how I have artistically captured or made an attempt to artistically capture this idea that your paper is putting forth. Yeah. Because I think
I like it.
Part of my style is to try to communicate sort of the emotion and to personify elements of biology, which people say you shouldn't do, but I do it anyway.
No, I'm very much in that boat too, to be honest, as an evolutionary biologist in particular.
Yeah, because there's a story behind evolution, right? Like the story of evolution is kind of the beating heart behind all of biology. So
Amen. Yeah.
when I read your paper, or especially just, reading the abstract, I had a sense that there is a notion that genomicists, I suppose is the right word, are missing out on, and that is the fact that, among the excitement of discovering genes that may have arisen sort of from the nether, that there is a real possibility that some of these genes have instead been abandoned by their relatives. And that's kind of the idea that we're going to be talking about today.
Yeah.
So how does that like strike your ear? Is that like an accurate, kind of read? Do you feel like similarly with this chosen topic?
I think I like that. I do actually really like the art. I like the ethos that it imbues. I think the one word that I might suggest a slight change to is abandoned. Because to me, perhaps you can unintentionally abandon something, but I think there's at least like a notion of intentionality there. And I think part of to me, the sort of impact in how boring the model in the paper is that it's a sort of it's like a maximally boring model that we test, one in which the genes that are actually related to these genes that now look new are sort of like chugging along, doing everything totally normally, not like trying to evolve really fast and abandon these genes. It's really just like business as usual. And even though nothing interesting is going on, the genes appear, exactly as you say, to be abandoned. So it's not like intentionally bad parenting. It's more like, passive neglect.
Maybe
parenting.
the word should be lost genes.
Yeah. Loss is good. Yeah.
All right. I like that. Okay, so here's kind of the vague outline of what we're going to be aiming to do today. First, oh my God, you know what? I was having such a fun and good conversation with you before I hit the live button that I forgot to do all of my social media things. So sorry. Let me real quick.
Oh my God.
Real quick. Sorry, VOD watchers. I need to do this. I need to tell my Discord I'm live. Okay. Discord will be sufficient. No one really follows me on Twitter anyway. Okay. All right. Sorry. All right. Back in it. Where was I? Agenda. Right. Okay. So I wanted to talk a little bit about my own history with this idea, because I want to emphasize how fortuitous it was for us to be having this conversation, for me in particular, as someone who is doing content about biology for a living, sort of my own pathway into your story. And then we'll have a quick review on different mechanisms of gene birth to kind of contrast more easily traceable, maybe, mechanisms of how genes are born versus this nebulous, popped out of the nether de novo gene birth. And then we'll actually take a look at the picture, which is the paper, which is going to be very exciting. And then if we have time, a little bit of a Q&A at the end. We have that to look forward to. All right. So the first instance of me actually thinking about like genetic novelty, this idea of new genes coming up, started with this video. Oh, and Witty am I asks, can you please explain the basic idea behind the paper? I wasn't able to catch it from the comment. Oh, no worries. The basic idea of the paper that we're getting towards is this idea that we should be and Cara, you can feel free to like correct me on this, but we should be gatekeeping what we call a de novo gene.
I love that. That's my new mantra.
Yeah, because I think when I was reading the paper, I'm like, oh, Cara is the gatekeeper. You are the custodian of what people get to call a gene that popped out of the nether.
I would be honored to have that title. Thank you for putting that into the
That's sick. All right. So it started out with this video that I made about the prion protein. I don't know, Cara, how much you know about the prion protein or the prion gene, but actually, it is one of my most beloved proteins. It's spooky, it's scary, it's got an incurable disease behind it. Who doesn't love the prion protein? But what people don't really know in the popular space is the fact that the prion protein has a shadow twin. This shadow twin's name is Doppel, appropriately for doppelganger, and this resulted from a gene duplication event of the proto-prion gene a very, very long time ago. I don't remember when. I think it was before mammals, but or around mammals, because I think fish also have a version of the prion gene. And this is pretty much the first instance of me going, oh, genes can happen. You know, a new gene can happen, which sounds silly. Like, I promise you, I went to school for biology, but I didn't really think about that as an idea in like a narrative sense, right? Okay,
Isn't that interesting? We never teach that. It's really true.
Yeah. You just kind of you get hooked up with Darwin's theory of evolution, you get hooked up with Mendel's pea plants, and then you just put them together and you just assume that new genes happen somehow, right?
Yeah.
But yeah, I was never really taught besides like mutation plus selection equals new gene. I'm like, okay, cool, and I moved on. But this idea of a gene duplication resulting in a new gene, I was like, oh, yeah, okay, that's a layer of detail that makes a lot of sense. And then I covered this experiment, the long-term E. coli evolution experiment, which follows this lab and their quest to just culture E. coli forever. I love this experiment. And what they had found in this 37-year period is one of their cultures, of their E. coli cultures, had a new gene pop up that allowed E. coli to digest or to take in citrate from their environment. So E. coli, they are famously citrate minus, meaning they can't use citrate as their sole source of carbon. And that is because they don't make the transporter too. They just can't, like, you can imagine it as like E. coli don't have mouths open for citrate. But one of the lineages actually did evolve this novel gene that actually enabled them to eat citrate. And the way that happened is through another gene duplication that actually stitched together the citrate antiporter, which was inactive in E. coli, and stitched it with a totally different promoter from a totally different unrelated gene to form this hybrid gene. And this hybrid gene is always active, enabling the E. coli to actually eat up that citrate. And I think that's really awesome. And in this lab, they've also discovered that E. coli have a lot of sort of proto-genes, this idea that they're making RNA that may or may not be made into protein, that are not really obviously functional, but that there's this bubbling sort of vat of RNA that could potentially be made into protein that really got me thinking about this idea of genetic novelty. And I'm wondering, Cara, if you've heard about this experiment or if you have maybe challenges to these ideas, or maybe this isn't as cool as I'm thinking it is.
No, I think this is very cool. I love this experiment. Like, a personal backstory is that I worked next door in grad school to a lab. It's not the Lenski lab, but it was Michael Desai's lab at Harvard. And there was a grad student there who I'm good friends with, who was Michael Desai and Lenski together published a paper where they did a lot of very high resolution sequencing of the Lenski line. So I have like 1 degree of separation from this experiment. I'm a big fan. I think that's a very good summary.
Oh my God, that's awesome. Like,
yeah, it's like a huge experiment in, like the molecular evolution field, because as you say, it's like, you're watching evolution happen. It's super cool.
Yeah, and it's just so funny. The guy, I had no idea you knew like the.
That's it's a small field. It's a small field.
Yeah. Yeah, and Wacky Lemon is also saying something that's correct as well, is that it's, DNA can lead to more things than just protein. It can also lead to RNAs that are functional as well. I don't actually know if E. coli have like fancy functional RNA.
I think they do. I think they probably do.
They probably do. I just can't remember. Yeah, I just can't remember if they do. Nothing obvious pops out to me. but yeah, this like exciting bubble. I think of like I'm gonna appropriate physics so bad. So if there's any physicists in the chat that want to correct me on this, but it kind of reminds me of like the edge of a black hole, where there's this like immense amount of energy that's creating like particles and antiparticles, and like an antiparticle will like fall into the middle of the black hole, but the particle will like spit out, and like this bubbling mass of foam that has infinite potential that could give rise to something real. I think is something that's very exciting about this idea of de novo gene birth. But as we're going to see, Cara is the gatekeeper, making us really think about like the standards by which we have to consider de novo gene birthing.
Yeah.
Okay, then the giant virus video. I really enjoyed making this video because giant viruses are so wacky, and I'm really curious to hear what your take on giant viruses are, if you have one. So giant viruses were once thought to be their own domain of life. Once thought is kind of controversial. There were people who were suggesting that giant viruses, see, now that there's a scientist in the room with me, I have to be very careful with how I word this, because in the video it's like,
I think you are a scientist. I will object only to that. But, yeah.
Okay, thank you, Cara. But yeah, there were people who were arguing that giant viruses could be a fourth domain of life, and part of their argument was the fact that these giant viruses have a lot of orphan genes. So ORF stands for open reading frame.
Great fun, right?
Yeah, it's a great word. For those of you who are unaware, ORF, that's open reading frame. This, I mean, maybe Cara, you can help me with this explanation, but I kind of view it as like, it's a stretch of DNA that has the paperwork to be a gene. Like, it's not confirmed to be like a real protein producing gene, but it looks like it could be a gene. Is that accurate?
That's a very catchy explanation. Yeah.
Yeah, it's got paperwork.
Yeah, exactly.
Yeah. And so what an ORF-N is, it's these things that look like genes that don't share any familial resemblance. They've been orphaned by the rest of biology. And so part of this argument was that, giant viruses are their own thing, is because they have so many genes that we can't find any relatives for. So, I have that as a bullet point here. Great. Good job, me. So if giant viruses contain so many genes that we can't find familial relationships, does that mean that they are special, or could something else be going on here? I'm wondering what your take on that is.
Yeah, I have not, I should say, in my like direct professional life, I have never done anything with viral genomes. I view them as sort of like their own beast. Things are much like viruses are sort of their own beast. They evolve much more quickly than animals, which are, or single cell eukaryotes, which are most of what I work on. So I have no personal experience, but because their mutation rates are so high, the paper of mine that we're going to talk about, one of the punchlines is that if you have a gene that's evolving very fast, basically like a game of telephone, it's just really hard to tell that it's related to some other gene. And so my sort of null hypothesis with all the orphans and viruses has always been that that's what's going on. But like speaking in my role as gatekeeper, I'm like a pained gatekeeper because I do think the idea, this is why I got into the field. The field of de novo birth is it's an extremely tantalizing idea. So my null hypothesis with the viruses is that it's just like sort of boring fact that viruses evolve really fast. But I do sort of want to believe underneath it all that there's something interesting there. But as far as I know, no one has really compellingly demonstrated that. So that's both my optimistic and my pessimistic take.
Yeah. And actually, I really wish I had this conversation with you before I made the giant virus video, because I was just kind of like, you can tell, I think if you're paying very close attention, that I'm very insecure with my argument for giant viruses not being a fourth domain of life. Like, I am personally insecure with that because I don't have, your background. But I was just kind of like, maybe they've been horizontally transferred from something that doesn't exist anymore. But I didn't really think about the fact that it could just be regular mutation that happens really fast for whatever reason in giant viruses.
Yeah, but if I recall, you do make an important point, which is that, right, it could be horizontally transferred. Maybe you mean from a eukaryote, but I think you also talk about the fact that viruses are just so undersampled that, like, if they're evolving fast, even if they're not evolving that fast, if you just haven't sequenced something at all close to them, that sort of makes especially problematic this problem of the sequence having evolved so much that you can't detect a relative. And so that, I think, is also a really important point that's very acute in viruses. You're totally right there.
Guys, Cara watched my video.
I did my homework.
Mylan. So Mylan is a fellow member of VTheorem, which is a group of virtual YouTuber science communicators that I'm a part of. And also, I actually asked Mylan for a little bit of help with regards to reading your paper. They're a statistician, actually, and I was struggling with some of the language. Not to say that the paper was, anything less than great. It's just that I haven't done stats in a while, so I was like really struggling. And so Mylan says,
Yeah, it's a little technical.
the method of finding that null seems so cool to me. I definitely have to check out the stats part of this paper because wow. And I do think that your way of thinking about gatekeeping the definition of de novo, I think it is also very compelling, even though you're kind of throwing cold water on the de novo people. But but for reasons that I'll talk about later, I actually think that adds like a delicious layer of nuance, I think.
I hope that's the conclusion. I would be very happy with that.
So I think you kind of answered this last question on this bullet point, which is this genuine genetic novelty or indicative of something darker, like the dark genome, which is like a term that I've heard.
Yeah, it's a good term.
Yeah, but like maybe, who knows, right? And in
Yeah,
fact,
I think until for me, the right view is until proven otherwise. I mean, this is sort of the punchline of the paper. Like, we show that there's an alternative, really boring explanation for these things. Like, they could be de novo, but we provide an alternative explanation. And sort of my personal view as a scientist that not everyone has to abide by, but I think is sort of good scientific practices. If you have a boring explanation, it's sort of good practice to assume that that's the case rather than jumping to the exotic explanation. But people in the field like actively disagree about that point of view.
which is also, I think, a lot what we do on these like journal club streams is I don't consider myself a teacher anymore. It's more about modeling ways in which you can interact with very complex information. And I think your philosophy I don't say that in like a demeaning way, but because science at this level is also philosophy. And that's what makes science so hard, actually. If science were just about like learning facts and like putting two and two together, it would actually be pretty easy. But at the edge of the darkness that is our sphere of reality that we know of is this philosophical quandary of like how do I choose interesting problems and how do I tackle interesting problems are things that you don't learn in a textbook. You learn it through conversations like this. So I do appreciate that perspective as well.
I love that. I started my degree doing a philosophy degree, and then I only later pivoted to science. So yeah, it's
Oh,
definitely that's why I'm in this field, I think, really.
that's sick. Because part of what you're doing is what is life? It is a pretty philosophical
Yeah, it's the big questions.
thing.
Yeah.
Okay, so this idea of how are genes born? I've been like these ideas have been bubbling in my head for a while. Like cryptic genes, orphans, gene duplication, sequence divergence, and that led me to make this video, which is "How Are Genes Born?" My most recent video. If you haven't watched it, please go watch it. I think it's really fun and funny. I think I'm really funny in it. Please watch it. But I'm not going to go too much into what that video was about. So, please actually do go watch it. But from that video, I wanted to review some of the more easy to explain ways that genes can be born. And I want to contrast that with this special case of gene birth, which is de novo gene birth. So as we said earlier, you can have a new gene pop up because you had a duplication event, replication oopsies, and then you get a lot of evolution, and then these two like twin brothers eventually become totally different, not totally different, but different enough that they have a different function. So that's one way of doing that. There's also gene fusion. Sometimes you have replication oopsies that just merges two genes. That's also possible. Or the other way around, you can have a gene that eventually breaks into two or in several pieces, and then that can be a way that new genes happen. Exon shuffling, this happens, in part in meiosis, where you have crossing over of different chromosomes, where you can. So now we're talking about exons and introns and not just genes. So this diagram is a little complicated, but imagine you're a eukaryote and your genome, you don't have just one block of DNA that becomes protein. You have a couple of different blocks that are stitched together to become that final piece of code that then becomes protein. So you can imagine like on this part of the chromosome up here, this is a protein that's made of three parts that look like this. And then on the other strand, there's a part that looks like this. And then you can have kind of entanglements that happen within these strands. And then when those entanglements resolve, you can have these these like DNA parts switch or flip. I think this is the hardest one to explain in like a casual point of view, but that's the best that I've got, I think. Okay, you have other ways. You can just get gifts from the outside. This is transposable element domestication and horizontal gene transfer. So you have parts of your DNA that can just pop out and pop back in. And if a mutation happens to fix that element, then that can become a real gene. By fix, I mean like prevent it from jumping back out of your DNA. Horizontal gene transfer, you can just have a virus come over and inject their DNA into you. This happens in certain diseases like HIV. And sometimes it's not bad. HIV, that's bad, right? But in our evolutionary history, there was a virus at some point that gave us the syncytin gene, which became important for, I think, implantation. It helps make babies, is what I remember from that story. Woody M.I. says, shouldn't exon shuffling happen only between alleles of the same gene? Gary, you can correct me if I'm wrong, but I believe that any region of homology, you can have a crossing over event. So it's probably more likely to happen between alleles of the same gene because they're more likely to be similar, but it's not like a hard cutoff, right? You can have similar regions of the genome that are in different places on different chromosomes.
Yeah, exactly. My newest preprint is actually about exon shuffling. So yes, that's correct. And we can, if anyone wants to, we can talk more about that later.
Oh yeah, in the Q&A, that'd be great. Yeah, I do want to get into what your next projects are in the Q&A as a teaser for the audience.
Yeah.
Okay, de novo gene birth. This is the thing that we're talking about today. I think it's super cool. I love thinking about this, and part of what we're doing here is reining in the excitement, because de novo gene birth is taking this area of the genome. That blue bar doesn't show up on the slide for whatever reason, but taking this region of DNA that doesn't actually code for any protein, it is quote unquote, it is non-coding and also probably quote unquote junk, which is a problematic term, but whatever. A region of the DNA that is never meant to be read to make protein. So there shouldn't be any reason why this area of the DNA does anything with regards to protein production. But lo and behold, a couple of mutations and pop out of nowhere, out of the ether, out of the nether, you have a brand new gene, which is an idea that I really love. But the idea of it is tricky, as we are going to see in just a little bit. So what separates de novo gene birth from these other mechanisms of gene birth is the fact that these other methods, gene duplication, gene fusion, gene fission, exon shuffling, transposable elements, horizontal gene transfer, you can control F for these, right? You can find these. If you suspect that a gene is the result of mashing gene A and gene B together, well, you can just control F for gene A and gene B and see if that's true, right? These are I'm not going to say very easy, but there are straightforward methods, I think, to find them or find evidence that that is how that gene was born. De novo gene birth, however, by definition has no homology. There's no existing familial relationship with a gene that pops out of the nether with anything else that looks like a gene. So there's a kind of searching problem that I think we're setting up here. Would you say that's accurate, Cara?
Yeah, absolutely. That's great.
All right. So we have, I'm going to say this word wrong every time I say it, but we have an epistemological problem, right? We have a problem with regards to the limitation of what we know and how we can know it. Because you can imagine that a gene that pops out of nowhere that has no familial relationship with anything else can be birthed in two different ways. They could be truly, and that's what this explanation is in this top left corner, they could have been just spawned out of the ether, right? If you play Minecraft, maybe you just put down a spawn egg. Like, you didn't have to have like a thing that came before. Or, and this is the more troubling explanation for people who are really in love with the idea of de novo gene birth, or did it have a parent, but did that parent, or a family, rather, and did that family drift apart faster than we could keep up with in terms of evolutionary history? Basically, was the familial relationship that this gene had, did that get wiped out over evolutionary time? And we just can't recover that relationship anymore. And I find this to be very compelling, because this is a hard problem, not just difficult, but this is like a hard limit testing problem. which is kind of spooky.
Yeah, yeah.
So we have this like philosophical quandary. It's so cool that you have had a background in philosophy, but I kind of think of this as like the ship of Theseus, right? Like, how do you tell if a gene is actually novel, right? So scenario one, de novo
Yes,
gene birth.
great. Yeah.
You just pop, genes there, okay. But how do you differentiate that from a long series of mutations that makes a previous gene look unrecognizable? It's a headache.
Definitely. Yeah, so there's the practical question here, like how do you in practice with given the finitude of our methods tell the difference? And then maybe what you're also getting at is like is there a point at which gradual mutation between two genes that used to be related, like if I get enough mutations, maybe it is biologically not meaningfully biological novel in some way and I can call it new. And so both of those are issues and I think especially after this paper many people who want to claim that these genes are new sort of fall back to the second thought, which is okay, maybe you can't say for certain that they have popped out of non coding DNA, but look, they're so distant in terms of number of mutations from their homologs that they must be doing something important. So both of these are live issues.
So I have a bit of a mantra that I say on stream and in videos too, and I'm wondering how this hits you here. But something that I constantly remind my audience of is the fact that biology is a spectrum. That like
Yeah.
our science is there in the language we use is there so that ape to ape we can communicate, but the words that we choose have like a finite information capacity because the ideas are infinitely complex. So I'm wondering how you respond to that kind of mantra.
Yeah, I think that's exactly right. The philosopher in me wants to say that not maybe not even a lot, but there's a substantial amount of confusion in biology. That's what Wittgenstein would call language games, right? We like use a word exactly like you're saying in some way, and it takes on some meaning or importance that's not actually inherent in the world, and then we just fight with each other a lot.
Yeah, like what is alive, what is not alive? I'm so bored. i'm I'm personally
Yeah.
bored of that question. I don't find that question to be that interesting.
Yeah, I agree with you. Yeah.
Yeah. Okay. Can we expand on that? Because I feel like people are surprised when I say I no longer care about the live or not live question.
Right. Right. There's like properties that sort of inhere in the world, like does it metabolize? Does it reproduce? And that is something that I can look at as clump of matter and answer yes or no to. Right. There's a sort of clear readout. But live is this like gestalt. Alive is this gestalt concept that we've sort of like sloppily and intuitively put onto stuff. And like when it didn't come from a clear definition of the world, we sort of shouldn't be surprised that we can't then make it cohere to a clear definition of the world.
Yeah, it's like vibes. Life is like vibes, I think.
It's vibes. Yeah, exactly.
And for everybody watching, this is I'm not the only biologist who thinks this way. Like it surprises people that a biologist wouldn't care about the definition of life, but I don't know. I don't find it very interesting.
Funny. Yeah, I'm with you.
Okay, very cool. Thank you for that validation.
Yes, strongly validated.
Okay. So we in my how are genes born video, I came across this de novo gene or a gene that people are calling de novo. This is BSC4. It's a yeast gene. It stands for bypassive stop codon, which is also like a really interesting idea of how biology has no boundaries, and sometimes a stop codon isn't even a stop codon. But that's neither here nor there. I just that's a cool idea. So this is one of the most well-characterized, earliest discovered de novo genes. It's something that people will label as de novo because, and you can add or correct any proof that I have, but it only exists really in Saccharomyces cerevisiae, which is brewer's yeast, and not in its direct, very, very close cousin, Paradoxus. Very cool name for a yeast.
Yeah.
It has no known homology to other yeast genes, so it doesn't look like anything else in the yeast family. So, what is your take on whether or not this is a real de novo gene?
I think I actually, if anyone is curious after this, I wrote a little review about like after this PhD work where I would, as you say, I gate kept everything and I believed none of the results. I wrote a little review summarizing all of the high confidence cases of de novo gene birth that I could find. And this one actually made the list. So I think it is one of the best examples that we have. And I can get into the reasons for that if anyone is curious. But I will say, and even in that, so the review is called Against All Odds, and it's in the Journal of Molecular Evolution. It's open access, which I paid a lot of money for, so it'd be great if someone would read it. It's really expensive to publish open access, so I'd love to take advantage of that.
And thank you for doing that. Otherwise, I wouldn't have been able to read your paper. So
I'm so happy to hear that. Yeah, it costs a lot of money, but we feel good about it when we can do it. But I will say, even in that paper, I write, there's tons of caveats. These are the best examples, but if I put my threshold at being 100% confident, I wouldn't have included any of them, frankly. So I think this is definitely one of the best examples. I will say my most recent work makes me skeptical of even this example.
ooh.
So I don't know if I had to put a number on it, I don't know, like maybe 60 to 75%. But it's definitely like sort of poster child, as you're saying.
So Thomas is asking if the paper's the origins and functions of de novo genes against
That
the
is
odds.
correct.
Okay, gotcha.
That is correct.
Yeah, I think I read a little bit about I did read a little bit of that paper to prepare for today, and I noticed that in your section for BSc4, I think you had decidedly not taken a stance.
Yeah, I try to be pretty agnostic there because,
Yeah,
when you write something like this, you're always like, in 20 years, will I look like a moron in retrospect?
yeah,
So, because, it's very hard, as you're saying, the whole problem is sort of very hard. So I definitely hedged.
yeah. And yeah, I was like, I see the hedging, which is valid. Which is valid. That's evidence that you're very careful. I appreciate that.
Thank you.
Okay. So even though BSc4 had its status as, it's like this poster child, that's the thing that I kept on seeing in these papers that I was reading for that video. And in my research, I was looking for any kind of challenge to this idea that this beloved golden goose of de novo gene birth, like whether or not that was ever called into doubt. And I came across this paper, which, not like a popular paper. You can see it's got 3 citations. But it did make me realize that de novo gene birth could be challenged to begin with, that just because scientists label something as a de novo gene doesn't necessarily mean that it is. So, I'm not a genomicist, so I don't know how compelling the arguments in this paper are. But I will kind of restate the argument, which is that the BSC4 gene in yeast could have arisen from a combination of gene fission and gene fusion, because there are parts of that protein that look like other proteins that also exist in the yeast sphere, I guess you could say. And I don't want to I'm not like, a news channel where I have to be fair and balanced and give every opinion out there like the same weight. But I do want to acknowledge that I did not have a frame of reference for any challenges beforehand. And the utility of this paper was, this idea can be challenged. So, even though I put a spotlight on this paper, it's not an endorsement. It's just this contributed to an evolution in the way that I think about the problem. So yeah, that's why your paper comes in so handily, because your paper actually does provide a standard that we can take a look at these new genes and actually really think if they're actually really from the either. I don't know if you have an opinion on, or even if you've read this paper before, if you have a rebuttal or a reaction to the argument here.
I'm very torn about this paper. I have read it. I've read it several times, perhaps more than I care to. I'm sort of strongly with you that it's a really important, strong, marginally visible, which is more than any other visibility of an opposing view in this field has. It's an important example of an opposing view, which is important because, as you say, I think there's been a sort of bandwagon effect where no one has sort of sufficiently challenged jumping to the conclusions that my paper tries to push back on. So I give it a lot of credit for raising the profile and considering the alternative view. I will say I think the statistics in that paper are just not there. It sort of uses and abuses the concept of statistical significance as a result of the control F homology search that you were talking about. So I think the general idea that there may be an undetected homolog for this gene is totally possible, but I think the particular examples that he pulls out are not statistically legitimate.
And here you can see at the edge of science, it's no longer like good paper, bad paper. It's more like this is something that my advisor used to tell me. I think this is probably applicable, is when you read a paper, you're not looking to tear it down usually. You're looking to see what can you learn from this paper. And so as someone who's not a genomicist, when I read this paper, it felt like it was like hypothesizing, and I didn't really have a background, so I couldn't take anything concrete away. What I took away from the paper is there is a route to challenge, which I thought was useful. So always, and this is just advice for anybody who wants to go to graduate school, because I fell into the trap of the second I learned critical thinking, all I wanted to do was point out flaws in papers. That's not really that's like an easy thing to do, is to point out flaws. The hard thing is to be like, okay, what is true about a paper? That's way harder, way more interesting than what is wrong with the paper.
Yeah, amen.
Amen. Okay, so uh okay, we can finally talk about your paper now.
I
Okay.
don't know, I was having fun, but I guess we can do that.
Okay, so paper. Many but not all lineage-specific genes can be explained by homology detection failure. So this is kind of a crappy slide. I pulled all this together yesterday, so apologies for this slide. But basically, this is like the top down. If you just tuned into the stream, this is kind of what you need to know. De novo gene birth, so gene birth out of nowhere from the ether from non-coding DNA, is a very exciting concept. It's so cool to think about. Oh, if you just sprinkle, some A's and G's and C's here, bang, you get a new gene completely. But we have scientists that are challenging the robustness of novelty as a concept. So that is the paper we are going to be reviewing, because not all new shiny genes might be actually new and shiny. They might be repainted, and we just will never know if they're repainted. So this might look like a biology as a spectrum, languages ape noise problem, which is something that I say a lot on this channel, right? Biology as a spectrum, because our the unit of language that we use is just doesn't have infinite information capacity. But in this case, it's not. Like, even though I've referred to the Shippa thesis before, actually this paper isn't really tackling that idea of like, oh, our language isn't imperfect. What you're suggesting in this paper is that there is a hard information theory limit to whether or not we can say that this particular gene has no family, which is a really complex idea that we'll repeat a couple of times, because it's very easy to look at your paper and go, oh, this is about, yes, this was de novo gene, or no, this is not de novo gene. It's statistically complicated. That's why I think statistics is like number philosophy, right?
Yeah, I love that.
Yeah, I say it all the time to my buddy Mylan here. It's like, yeah, because is it math? It is numbers, but it's not like math usually has an answer. 2 + 2 is 4. But statistics is an argument. It is philosophy. And so what we're encountering here is that there's a hard epistemological limit to our confidence as to whether or not a gene has family members. Is that an accurate way to set your paper up?
Yeah, absolutely.
Awesome, sick. So part of doing this is I was talking to Cara about this before starting stream, but like, I'm really happy to have a scientist on, because I so rarely get told no, and just the idea that Cara could be like, you're wrong, I'm like, that means I get to make sure that I'm still thinking in a sharp way. So I appreciate that. Okay, so here is sort of the two scenarios that we have set up here. There's the null hypothesis, and this is what I asked Mylan for help on, because it's been a while since I thought about the word null hypothesis. And this is the part that I'm most insecure about. But the null hypothesis, I think, is that the gene isn't really de novo, or rather, it's just as likely to be a failure to find relatives. And then the alternative hypothesis is that that's not the full picture. The de novo gene status cannot be fully explained by a failure to detect homology. So this is kind of two jelly stances, I would say. Like they're pretty soft and squishy as robust stances to take on the opinion of whether or not a gene is de novo. But it is hard in the fact of saying, like, I'm getting wrapped up in my own head, because this is kind of, sorry, maybe you could take the wheel for a second as my brain kind of unclutters, because it is a little bit tricky to separate these.
Sure, if you like. Yeah, but you should jump in at some point if you wish to redirect. It's literally your show. So, Okay, yeah. So, right, to me, in general, as a biologist, but a sort of like vaguely statistically minded biologist, the idea of a null hypothesis is really important. So usually in biology, we're interested in finding some new shiny effect. Here, we're like interested in this super exotic hypothesis of de novo origination. So that's what I'm going to call the alternative hypothesis, exactly like Phi is saying. So that's like the interesting thing that we're looking for. And we use a statistical test of some sort to assess whether that's true or not. And that's this control F homology search that Phi was talking about earlier. But when we do any statistical test, we want to in the mathematics of the statistical test, which maybe Phi will talk about, but if not, we can sort of schematize. you want to be testing, you have some number and you're doing a statistical test, and you want to see whether there's a high enough statistical confidence for the alternative hypothesis as opposed to something else. And that something else is the null hypothesis. And so here, exactly like Phi was saying, the null hypothesis is actually there's absolutely nothing interesting going on here. It's just what we're calling a failure to detect homology. And so the cute little graph that Phi has schematized at the bottom shows a cartoon representation of what we might think of as the statistical number that results from this control F search. So if I hand you some protein and I'm like, okay, I think maybe this is a de novo gene, how would you actually go about testing whether or not that's true? Like if I said you would want to see if it looks related to any other protein that we've ever sequenced. So you would do some control F in that very large database. And what that machinery is doing, this is an algorithm, a general class of algorithms called homology search. And you can think about it really abstractly as basically trying to see how similar to sequences, by which I just mean strings of characters, where the characters are like ATGC, or for proteins, they're the 20 amino acid characters, how similar those two sequences are, and statistically quantify how likely you would be to get two sequences that similar by chance. So if you're higher on this scale, you are more confident that they are actually related. So if had, for example, in English, if I had two sentences that were absolutely identical, you'd be like, okay, well, I wouldn't get that by chance from two monkeys typing on a typewriter. Clearly, that's actually the same sentence. And so that's really high on the x-axis. So that's like the blue point. But as, those sentences, now we're thinking of evolution, as those sentences mutate and different characters in the sentences over millions of years accumulate mutations and start to look different, eventually the crucial intuition is they will pass some threshold below which you won't actually be able to statistically tell that they were originally the same. And so if that
Yeah.
was like a little bit too statistically complicated, I think the telephone game that we all played as kids is a good example of this. Like you start with one sentence and you pass it around to your circle of friends, and everyone like changes a word. And eventually when it gets back to you, you're like, I actually have no idea if this was the thing I originally said, or if someone sort of like tampered with it intentionally along the way and like inserted a totally new sentence or not.
Yeah, Mylan's
And
saying
so,
like running a sentence through Google Translate like 100 times.
yeah, exactly. Introducing some weird source of error. Right, exactly.
So
So
in
when that happens, go ahead, sorry.
in your particular test, I yeah, as you interpreted my schematic, there's like a threshold that you are trying to calculate, where below that threshold it's boring, and above this threshold, okay, we can talk about whether or not this is de novo now. So it's not as simple as yes or no. It's more like we can't tell, or this is worth looking into. Is that a fair characterization of this threshold?
Yeah, exactly.
Perfect. All right.
Yeah.
Okay, so yeah, so we're gonna talk through this Google Translate thing, and we'll illustrate this. Why might a sequence fail to have detectable homologs? So this might seem pretty intuitive, but I just wanted to show you all what this would look like for a sequence of DNA. So this is two, you can imagine this as like a duplication event sometime in history where we have this gene, it's got a certain length, and they're identical at the time of duplication. And, a couple million years or whatever pass by, and maybe this gene accumulates some mutation, this gene accumulates some other mutations, and we start seeing the statistics on these change. So in this first, actually, I should walk through this a little bit. So I don't fully understand how these statistics work, but from what I understand, the e-value is what is important when determining whether or not two sequences have homology. And if you have an e-value of lower than 10 to the minus 10, then you're probably homologous. If you're in between 10 and 5, then you're maybe homologous. And then above 10 to the minus 5, you're probably not homologous. So this is, again, statistics is numbers philosophy. This is giving you a level of confidence whether or not two genes are related. And you can tell for this length of identical DNA sequences that this e-value is like omega tiny, right? Minus 10 to the minus 17, right? Super confident that these two genes are related. And then, yeah, if we role play like a couple million years happening and I've substituted some, like I substituted one here and one there, you can see that this E value starts cratering. Right now we're 10 to the minus 13. Still pretty sure that these two are relatives, which is cool. But then, you know, a couple million years later, and maybe there's some deletions in this sequence, maybe there's some insertions in this sequence, maybe some more things get changed, and all of a sudden your algorithms start being less confident that these two are related. So now we're getting into fishy territory of 10 to the minus 9, and then at some point the algorithm just decides that they are not related. And I'm actually really surprised that the BLAST search doesn't see these two as homologous. I'm wondering what your take on that is, or if I've just done these alignments incorrectly.
Well, I can't I don't know if I can like swear. I can't mentally compute that well on
Yeah.
a blast string, but it seems plausible to me. I mean, these are really short sequences, so even if the like percent similarity is high, the total information content, so to speak, in a short string is it starts out sort of not that high to begin with. So it sort of doesn't surprise me that much.
Gotcha. If I had chosen a longer string, maybe it would be a little less surprising why this
Yeah.
one doesn't actually return a meaningful comparison. But where your paper is going is that maybe this process of accumulation of mutation is happening so fast that we can no longer productively tell whether or not a gene is new or just really, really, I'll call it corrupted, because I like that imagery.
Yeah, exactly.
And this is also just my little soapbox because people keep on telling me I'm wrong in my Prion video. Yeah, I make the argument that it's unusual that the prion protein in mammals is conserved so well. And I say that, you know, this is indicative that there's likely something useful that the prion protein is doing. But I keep on getting told by people like, oh, past breeding age, you know, selection doesn't care about what your genetics are past breeding age. And I'm like, that's true, but we're not carbon copies of our parents. Mutations do accumulate. And if there was no selective pressure on the prion protein at all, then you would expect there to be some funky variants of the prion protein. But across mammals, the prion protein looks pretty similar across the board. Although there's like one weird mountain goat that doesn't make it. So kind of bizarre.
Yeah, that's cool. Yeah.
Yeah. But yeah, so it is a little more nuanced because I think in popular education it's like, oh, you know, genetics only matters up until you pop out a baby, but it's just yes and no is what I would say there.
Yeah, especially in humans, right? We have so much like grandparental care in our history. I always think about that as a selection pressure.
Yeah, like the pressure to have big villages that help everybody.
Yeah, exactly.
Although I didn't want to make that argument in my counterpoint, like because I did make a response video on my second channel. I was like, I'm not gonna get into Evo psych.
Yeah, that is dangerous.
It's dangerous, but I do believe what you're saying. I'm not ready to make that argument to people.
Yeah, same.
Okay. So you refer to this as a simple model, which as a non-statistician, I eventually, I think, figured it out. But I was like, oh, okay, simple model. So this is a mathematical calculation that you are modeling to determine two genes' similarity score. So the idea here, the angle that you're taking, is we can track the distance between two genes and two species as a metric of time based on how evolutionary related things are. And I'll show a graphic in the future. This is just to like give the vibe of this math down. So the first step is in order to check whether or not two protein genes can be regarded as homologous, you check their similarity score. Not going to get into how that's calculated. I dropped out of my comp bio class. I'm sure I learned how to do it at some point. And then you check their e-value. How likely is it that these genes are just kind of accidentally similar, right? You mentioned monkeys and typewriters, which is a phrase that I use like 10 times in my abiogenesis script. There's a lot of biology that is just cool. Monkeys and typewriters.
Yeah.
And then to determine whether or not a gene is really like brand new or just a failure to detect changes over time, we need to model how a sequence would naturally degrade over evolutionary time. So there's this assumption that you make in the paper, and I don't have the background to challenge your assumption, but I'm wondering kind of what went into your assumption that over time, a gene is just going to degrade at some some kind of rate. And if your
Yeah.
gene degrades much faster than these rates typically go, then you might not be able to tell if something is new or not, if it's just so blazingly fast that you lose all that information.
Yeah. Either blazingly fast or just it's been happening for like slowly, but for a really long time, which like in animals, we've sequenced like, I mean, maybe not all groups of animals, but in a lot of animals, for example, in humans, we've sequenced a very closely related animal. So if I don't see a gene in a chimp, it's probably not that it's evolving really slowly, but for a really long time, because chimps are very closely related. But this is relevant for your virus example, where there may be, like many, many, many millions of years between your virus that you sequenced from some random sample and the next closest thing. So either really fast or just for a really long time.
I do want to point out some fun comments. My brain just wants to like, this is Turnitin, but genetics. Gene plagiarism.
Plagiarism. Oh, I see.
Turnitin being like the plagiarism software that teachers use to check if
Oh,
students
I
have
see.
copied.
That's funny.
Okay, so math time. This is actually pretty familiar. I remember seeing formulas that look like this, but I actually, when you said simple model in math, I was like, oh God, I haven't done math in a while. But actually this, if I'm understanding this correctly, this is actually not that bad. So
I think it's so elegant. There's like two parameters. The context for this field is that people had sort of tried to do this sort of modeling before. Like, can I model the rate at which the sequence is changing and therefore that it's going to be harder and harder to see that it's more similar? And like when I came to the field, the way that people were doing this was with these like just brute force simulations of gene evolution in the computer where there were like, hundreds of parameters. So I was really happy to come out of this with a 2-parameter model.
yeah,
To me, that's like super simple.
I also appreciate that as someone who is deathly afraid of math. I read the paper, I found the explanations to be quite good, and I am putting my membranes on the line because I'm going to try to explain it to you. Because there's stakes to this, right? I think it's fun that I try and do this kind of thing. So on the left-hand side, we have that similarity score that's so important to establish like a baseline as to whether or not two genes are related. This is a function of time. So similarity is a function of time. And on the right here, the similarity score is going to be a function in part of the protein length. So longer proteins will scale in a certain way with regards to similarity. Shorter proteins will scale in a similar way. And then we have the exponential decay constant E, which does fancy things when you do this to it. Again, I'm bad at math, but I do remember E being a thing in exponential decay models.
Yeah,
and this constant I find to be fascinating, this minus R term. So R, you mentioned, is a protein-specific mutation rate, and you make it clear that you're only counting substitutions. So by substitutions, what we mean is, if a gene is AAGGC, that first A mutates to a G or something, like, swapping base pairs, essentially. And what struck me was there's no... Okay, I'll walk you through my initial emotional reaction. Is I read that this is the model and I went, ain't no way. Ain't no way it really is that simple. And I think that a lot of that surprise comes in this one parameter, this R parameter. So this is assuming, correct me if I'm wrong, this is assuming that similarity will decay in part, just statistics, right? Things will just fuddle over time. But my initial, okay, this can't be true, because what about selection? What about the fact that certain genes are important and less important, and when you do mutate them, you get a penalty? And what's crazy about this thing that you have chosen to model is that this R term just kind of sweeps all of that up in a neat little basket. Exactly. Oh, that's so cool.
Yeah. But I think your initial reaction is right. Like I remember very clearly the moment that I tried this. This was after this is like what a PhD is like. This is after like 2 years of trying these really heavy simulations and being like, this still isn't working that well and it's really computationally intensive. And then I just like drew this plot and just on a whim, like fit this 2-parameter exponential. And I was like, this actually looks really good. So I totally share your skepticism. But yeah, it does seem like selection, for example, can be incorporated into this parameter. There's a formal derivation of this model in which you have to ignore things like insertions and deletions. And as you say, only deal with substitutions. But it seems like, you can sort of subsume even insertions and deletions into this. I worked with this really smart grad student who was very stubborn once, who wanted to take into account that like the rate of evolution can change over time, and like so maybe it starts out fast and then at some other period in evolution it changes. So you might like think about two phases for the exponential decay. And I did try that at some point, but like it seems like to zeroth order, all of that complex stuff, just as you say, can really be bundled up into this one neat little parameter. And I always prefer simple models when you can get away with it. So I was like,
so
I was
sick.
surprised, but I was happy to leave it there.
Yeah, I bet you thought you were really cooking when you were drawing those graphs. Like
I know, I was
you
like,
were
can this be
cooking.
real? That's great.
We got a super chat coming in from Joey's. Just saw that y'all were live. Recently found you, Fi. We will watch your career with great interest. Thank you for making me. Thank you for making the time to come on, Cara. Keep up the mind-breaking work, both of you. Thank you so much, Joey's.
Yeah, thank you.
Yeah, this is sick. Like this idea that you can paper over all of selection by just like so for context, right? When people see a math formula, they say, okay, well, what are you putting in for that number? It's not about knowing what this number is, because as we'll see, that number itself, the actual value of it is not important for a gene. It's the fact that you can model change over time with this formula. So even if that didn't make any sense to you, just this is the takeaway. The similarity between two genes can be, I'm not claiming that it is exactly what. Okay, so this is another mantra: all models are wrong, right? Including Cara's right here. This is also a model that is wrong, but it is useful. All models are wrong, but they are useful. So you can use this formula in order to map out the mathematical relationship between two genes over time. So don't worry too much about, okay, what about selection? What about this? What about that? What you should take home right now is there is a simple math formula that is just a very neat exponential decay that simplifies a lot about evolution, sure, but it's also pretty useful with regards to defining a hard limit for whether or not a gene can be considered de novo. So I think that's very, very, very cool. Oh, I'm so glad that I actually understood that, because I haven't looked at a math problem in a while.
Exactly right. Yeah.
Great. Also, compliment to your writing, right? For me to be able to read it and understand it as someone who has not looked at a math formula in a while, it was well
Thank
written.
you. There was a lot of arguing about how to write this paper, so I'm very happy to hear that.
Yeah, if a dummy like me can get it, like that's huge props. Especially a dummy that doesn't like. I refer to myself as a dummy just for the sake of like, math was not my thing. Like I am actively afraid of math, and I was still able to get it, which I am happy. I'm smarter than I expected myself to be in this particular regard. So I was a little nervous.
I suspect that a lot of just is like a meta comment to get on my soapbox. I teach a computational biology and statistics class to freshmen. And like, as a little soapbox, I think so many smart people just have like learned this math phobia somehow. And I don't know like what sort of trauma you guys have had in your past, but like, there's no way that people as smart as you cannot do math. It's just like, I'm not making light of the psychological barrier, but I do think you guys are all capable of it.
I actually, this is not to get like too heavy into it, because I've like come to terms with like where my strengths and weaknesses are. My dad was like a math PhD, so like,
oh yeah,
yeah, he was very good at math. But we'll move on from there. Like I do, but I'm not. But the thing is that I have that like math anxiety, but I will rise. I will try. That's the important thing. Is that I am equipped with enough self-worth that I will at least make a crack at it. And I'm glad that I made the crack at it, because if I didn't, this would this would have been tough to cover your paper.
Yeah, well, it's perfect.
Okay, cool. So, using a comparison, so you picked yeast and Drosophila. I'm going to focus on yeast. You can talk about maybe a little bit why you chose also Drosophila a little later, but it looked like most of the paper was yeast. So for the sake of brevity, I chose to stick with yeast. So you can actually map out the distance between species as some measure of time due to how closely related these species are. So if we look back at this formula, like this time function, like this t value, you can insert a closeness, because you can measure the relationship between two species of these yeasts or these fungi as a measure. Sorry, one more time. You can map out the evolutionary distance as interpreted through time by comparing these species. So Cerevisiae, this is our guy that we're looking at, brewer's yeast, and he's got a very close relative in Paradoxus. So you can see that the distance is very, very short, right? That, and distance and time are related conceptually. So that is where we're getting time from. So we're not literally measuring like with a timer. This is how much time has elapsed. This is like inferring time because Cerevisiae and Paradoxus are like first cousins.
Yeah. And this is like a confusing thing about evolutionary biologists. We measure time in like number of DNA substitution. So like, I don't know how many millions of years yeast have been evolving. There's no fossils. So instead I just like measure on average how many mutations have happened. And then I'm like, I will give you all of my time in those units. So we avoid all sorts of complex issues by doing that. So that's why the numbers look sort of weird here.
Hell yeah. Hell yeah. As someone who's doing an abiogenesis video now, I wish I could just paper over time in that kind of way.
Yeah, it makes things easier.
Yeah. Oh, also, I just noted this because this freaked me out when I was reading this chart. S. pombe or whatever, this is not Saccharomyces. This is schizo Saccharomyces. So this is like not related. This is not the same family as brewer's yeast. It just happens to share the same first initial. So this guy is like very distantly related to brewer's yeast. And then as you go down this, the more closely related that you get to brewer's yeast. Okay, now we get into stuff that I find to be quite fun. I mean, I think you also downplayed how interesting some of this stuff is, but as someone who isn't a genomicist, like working through this puzzle was a lot of fun. So we'll focus on the left here. This is brewer's yeast, and we're tracking 3 different genes over evolutionary time. So time 0 is brewer's yeast, Saccharomyces cerevisiae, and then like long time is whatever a nid is, which I don't know. And we're tracking 3 separate genes. So we're tracking POL3 in red, we're tracking DUF1 in blue, and we're tracking MDM10 in rust. I want to say this color is.
I like rust. Yeah, that's a good name for brown.
Yeah. Oh, yeah. Actually, I guess it is brown, huh?
No, yours is better. They're
So
sort of arbitrarily chosen colors in retrospect, but that's fine.
they are red-green colorblind friendly, which I do appreciate. Not every biologist remembers that those people exist.
Yeah.
And so here's the power of this simple model, right? These dots, these red dots, these are the actual data, right? These dots are the similarity score per evolutionary time, right? So these dots are those scores. So when you take a look at Cerevisiae and you take a look at a close relative of Cerevisiae, it should be very similar, so it should be very high. And then when you come all the way out here to whatever this fungus is, the similarity score is very low, so that makes sense. And then when you plug in this formula that you had conceptualized beforehand, you get a very, very neat line through all the data. Curve, I should say. Like, it's incredibly
I feel that way
neat.
too, and no one will ever believe me, but these examples, I literally did just like randomly with a little random script in the terminal pick 3 examples. So this is really a purely random sample from the data.
It is.
It's a beautiful fit.
Shocking. Okay, so why is this shocking? I'm sure anybody with a background in statistics can tell. These dots are so, remember, this formula, this is what generates that line. This generates the line. And when you use the math formula, you can tell that the line captures the relationship of the dots very, very well. R square is a Mylan's also in chat, so I'm gonna get this terribly wrong. but R squared is some metric, some measure of how well does your math formula line up with your actual data. So how, like, if you're doing a line, the R square is like how well do your points lie on that line. This is a bit more complex because this is a curve. And dude, these R squares are crazy. Okay, so the maximum this can be is 1. If your R squared is 1, then all of your points lie exactly on that line. And the fact that these are like less than 10, like less than 0.1 away, like that's insane. Like, I don't have a concept of how rare that is.
Yeah.
Like,
amazing. This is part of why yeast are great. Like, as you'll see, the Drosophila fits are not quite as good because like animals are more complicated. But the yeast,
animals.
beautiful.
Like, how rare is it in the field of genomics to have a fit that good? I mean, give some context. Celebrate. That's crazy.
I was pretty happy. Yeah, I think it's pretty rare. I mean, it is a simpler problem than many things in genomics, but it is pretty rare. As I said, like given the simplicity of the model, I was extremely surprised to see this. Yeah.
So in this model, you are capturing issues like selection, but I would say like
Great.
the R is papering over selection, which I personally, from what I understand, is fine. I think the point that people might take issue with is the fact that there's only substitutions. But if you do just substitutions, I mean, this is good, man. Like this shows that you can capture a lot of the difference in similarity score just by paying attention to substitutions. I don't really know if there's going to be a lot more that can be explained by the more complex mutations, at least from my naive perspective.
Yeah, that grad student I mentioned, like, he is right that there are some cases, especially in animals, where it sort of looks like a two-part exponential fits better. There are some outlier cases, especially in animals. And the intuition there is sort of that, like, at first the protein evolves at some rate, and then sort of like there's some fraction of sites in the protein that are really important and really have to maintain their identity. Like perhaps in an enzyme, amino acids like right around the active site really have to be one specific amino acid. So you sort of mutate all the sites you can, and then some other fraction of the sites, preserve their identity. So you should have like a two-part exponential. So I do believe there are genes like that, but I think they're a minority. And like I said, I just prefer simple models. So
So
I was happy enough with this to leave it.
here's a numbers philosophy challenge to this data though, because I'm going to be not critical, but yeah, critical. At what R squared would you say that it's not a good fit? Because I don't have that context.
Yeah, I think like so one of the reasons that we showed this figure was like just to convince you that this stupidly simple seeming model actually works. But I think your point is well taken that like this is a, as you were saying, it's a spectrum, it's a continuous number. And so if you want to benchmark when your model is good enough, you want to see how it does on the actual downstream task that you're meaning to use it for. And so there's a figure later, I don't know if it's important enough for you to have shown, but there's a figure later where we do a sort of control on that task, which is we take genes that we actually can detect in the farthest species, so like a cerevisiae gene in Pombi, and we say, okay, so I know that it's actually there, but if I sort of remove that data point from my test and use the model to predict whether it's there, which is what we're doing for genes that look de novo, that we don't actually know are there. So we're sort of doing a test that's akin to what we're going to use it for. Can we predict whether or not it should be detectable there? Do we get the right answer? And so for this model, you'll see that that sort of like predictive test is extremely high accuracy. So I think you're exactly right to say like there's no R squared that I should be like, okay, this is good enough. I think that's measured in the downstream test.
Fantastic. I do believe that I do have that figured down there.
Cool. We can talk about it when we get there. Yeah.
But what we can say, at least for now, is that this simple equation does map similarity to time very, very well, judging by the R squared. So there is this relationship, and this sets us up for the gatekeeping tests that we're going to talk about soon. So this is my very simple Photoshop. I just drew things to help you understand the data that is about to come. So, right, we've got this relationship, similarity and time. So same graph, similarity and time. There's a certain threshold where you can't really be confident that you can detect a real similarity. So if we go back here, I don't know where the similarity threshold would be.
It's all the way at the bottom. It's really low. It's that dash line way at the bottom. Yeah,
oh my God, it's right there, Chad. I don't know if you could see that. I see. Oh, that's low.
So this really drives home the fact that most genes, like you're never gonna have any trouble detecting them. The number of genes that like might be de novo or might not, it's actually a really small fraction of all genes because most genes just like blow that threshold out of the water.
Gotcha. Oh my God, I really didn't even see it. I didn't even know you put it on the graph. Okay, that makes this explanation even better because, yeah, below this tiny threshold, this is the Shipathesis problem, right? This is like, if you dip below this similarity score, well, we don't know if you're a relative or just something else completely different. So if your curve between similarity and time dips beneath this similarity threshold, this is the dubious. This is the nether. You are so different that we can't tell if you are a relative or if you're something new. Like there's no meaningful conclusion that you can make based off of your sequence. However, if you have a curve and it's like pretty well above this threshold, if there is a relative, you should be able to find it. Is that accurate?
Yeah, exactly.
Okay, very cool. So I kind of view this from a philosophical perspective, because I like thinking about things from different angles. This is kind of reminds me of this conundrum with regards to the observable universe, right? So if you, and I'm appropriating physics, so please tell me if I'm wrong. But, here we are, blue little marble in space, and there is a finite layer of, like an opaque layer that we'll never be able to see into. No matter how good our telescopes get, there is a hard limit to how far we can peer into the universe, because the universe is expanding and that expansion is accelerating, meaning that because light travels at a constant rate, we will never see the edge of the universe, no matter our technology, because those photons will just never smash into our telescopes. And so this presents a hard problem, not a difficult problem, because difficult implies that it is solvable. This is an impossible problem, right? Light from the edge of the universe will never reach us. It's too far away. It is getting further away, and light cannot speed up. It's already at the maximum speed. And in the same way, I think about detection failure is the same way. Like, these sequences have mutated beyond anything that you could scrape back into the way that you think about things, right? Like, the information is lost. Not hard to recover, but philosophically lost into the void, not recoverable. Like, we cannot say, because of the ship of thesis, idea of like, your sequence is just too different. Well, we can't tell if you're related to anything. How does that read to you?
I think that's right, with the caveat that I think that's 100% true for the particular Control F homology search method or class of methods that we built this model about, which are like, I would never inflict on you guys the derivation of this, but it applies specifically to models, to search algorithms that only use the information in what I'll call the primary sequence, which is like the linear, arrangement of letters in the gene. And so like that, the test like BLAST that uses that information definitely has this fundamental limitation. But now, like, you guys have probably heard about AlphaFold, which is a structure prediction deep learning model. And so some people are now applying that to the problem of homology search. So the idea is I give you something that looks, I think in your de novo gene video, you predicted the AlphaFold structure of some like random peptide, right? And it can do that without knowing any homologs. So some people have started like predicting the structure of a de novo gene and then doing like a structural comparison, like a three-dimensional point-to-point structural comparison of de novo genes to like the structures of known genes. And when they find a hit where the structure looks similar, they're like, okay, that's a homolog. And so the short way of saying what I'm trying to say is that there may be like sort of radically different homology search mechanisms where, because the information that it's relying on is different, like you could maybe sort of imagine that a protein over all of evolutionary time maintains the same structure. And if you can capture that information, then maybe that's not sort of fundamentally, irreversibly lost over time. So I think there's some hope. And I say this because I did my PhD in a lab that like writes the software, different sorts of software to do homology search. So there is a little, core of homology search software makers out there who are hoping to push the bounds of this fundamental loss of information over time. So we hope that they're not totally immovable, but definitely for this sort of technique, which is pretty basic as far as they go, this is 100% true.
Yeah, get on this. If you're a student, get on this. That sounds great. That's awesome. Witty MI says, shouldn't the gene suspect of being de novo be outliers of the exponential trend? Oh, interesting. Keep that in mind.
Yeah, that's a great intuition.
Keep that in mind. Hold on to that. So you have the mathematical formula. It models the relationship between similarity and time very well, at least for the 3 random genes you picked out of a hat for yeast and Drosophila. Now we're going to look at some candidates for de novo gene birth. So ULI1 is a candidate for brewer's yeast de novo gene. And so the null hypothesis here is the gene is just as likely to be de novo as it is to be a failure of detection of homology. This protein is not an interesting candidate for de novo gene birth. The alternate hypothesis is that the gene is not likely to be just a failure of detection of homology. It's more likely to be interesting. So just reminding us of the dichotomy that we're setting up here between the hypotheses. So this is what this curve looks like. So obviously, to walk through this is a gene that is only found in Saccharomyces cerevisiae. So that's why there are only 4 points on this line. But we trust this model right? We trust this math because we saw proof that this math worked before. So even though we only have 4 points, this line still maps beautifully to these points. But something interesting happens when you go far enough where you're no longer talking about brewer's yeast. Your curve dips under the detection threshold. So it collapses very, very quickly. So do you want to talk about kind of what this means for ULI1?
Yeah, sure. So yeah, exactly. It's this. I'm really glad you emphasized that we trust this model because that's why we showed you those plots before. So you trust this model, and the red line is basically saying if this gene is evolving in exactly the same way after the group of 4 yeast where we detect it as it is there, what would we expect its similarity sort to be? And here you can see the dash line, which is this detectability threshold, below which you basically can't tell anymore whether or not it's really related. So the idea is if the prediction for this value falls below the line, even if the gene were there, we would not be able to see it. So you can see the red line is solidly below the detectability threshold. And then something that we didn't talk about mathematically is that the red line just gives you a point estimate. But there's like uncertainty in the modeling, and the evolutionary process is stochastic, not deterministic. So you can model the uncertainty around that, and that's the sort of purple cloud. But even if you take into account all of that uncertainty, there's a 100% probability, according to this model, that if the gene were evolving exactly the same way in the first species where you don't see it, even if it were there, you wouldn't actually be able to see it. So this is saying, okay, we find that it's not there, but we actually don't expect to find it, even if it is there. And therefore, the inference that it's de novo based on not being able to see it to people like me does not seem very well founded.
So this is kind of like the gatekeeping problem, right? Like
Yeah,
even
exactly. Now I'm going to gatekeep. Exactly.
even if Saccharomyces, I don't know what this is, Cass, even if this did have a relative, you couldn't find it because it's below the gene, the similarity threshold. Like,
Right.
because that's where this red line is to fully spell it out. And I think that's dope. That's a very neat argument, I think, for
Thank you.
gatekeeping some of these. And
Principled gatekeeping. Yes.
yeah, yes. And then we look at another candidate. So this is SPO13. So these genes don't have structures. These were folded by AlphaFold. If you've ever folded
That's
things in
cool. I actually
AlphaFold,
haven't looked at this before. That's cool.
it's really wacky, very
Yeah.
disordered kind of protein, according to AlphaFold, of course. So again, the null is this gene is just as likely to be de novo. So this is the uninteresting protein hypothesis, and then there's the alt hypothesis, which is that this is an interesting protein. And this curve looks very different. This curve decays a lot slower over evolutionary time, and only when we get out to whatever the heck this guy is, do we see that decay even approach the detectability threshold, which means that if there is a relative for SCAS, right, it might be somewhere around here, which is above the threshold. If it exists, right? If there is a relative, we should be able to find it, is what I'm taking from this. Is that right?
Exactly. Yeah, exactly. Yeah.
And then I tie this back because when I first read the paper, I I had the attitude of like, oh, you're gonna gatekeep yes de novo or no de novo. But when I looked, after reading the paper and taking a look back at the title, I'm like, oh, you've been very careful since like the beginning of the paper because you're not actually gatekeeping here in terms of like hard yes, hard no. You're just saying we need more evidence, which is a different thing than outright rejection. Right, some lineage specific genes are poorly explained, cannot be captured by this math, or like not a convincing argument. Another hypothesis is possible, right? This very careful threading of the needle to not actually hard gatekeep, but like soft, as you said, principled gatekeep.
Yeah, exactly. Yeah. So, and actually, I can't remember if this is one of them, but so in general, when you're doing this, testing an alternative hypothesis versus the null hypothesis, sort of formally what we're doing here is like we have an alternative hypothesis in mind. And the way you've put it is exactly the right amount of vague. It's like, the null hypothesis can't explain it. But just because we've, to use technical sort of terminology, rejected the null hypothesis, I've shown you a very simple model of like simple evolution and showed that it can't account for the fact that we don't detect this gene here. There could be anything else not going on in that model that causes it to deviate from the null model. So it could be de novo, It could have just changed its evolutionary rate between the species that we detected, which are all the little black dots, and Castelli, which is S-Cas. So maybe it like started to speed up because it's like the same gene, but it gained some new function. That's also one possibility for why you might not see this. So there's like a whole suite of explanations that might cause this, not just de novo birth. And we don't have the power to test that with this model.
And I'll just spell out the math one more time for people to. So these dots above S. cerevisiae brewer's yeast, these are sequences compared to S. cerevisiae, okay, so this is like comparing the SPO13 sequence across these several different species of yeast that are closely related to the reference S. cerevisiae. And so this is our like data. This is like the actual similarity score between different SPO13s that you can find in this S. cerevisiae family. And then the line through that is the math formula. You take these dots, you plug it into the formula, and then you let the formula generate the rest of this curve. That's what we're actually looking at here. Okay. So cool how like it was a neat like intellectual exercise to be like, oh, how do we actually gatekeep this? It's not just like, oh, you reached a number and you didn't reach a number. It's like, oh, the shape of these curves is what is gatekeeping.
Yeah.
That's cool. Above or below the curve. If there's anything you take away from this data, is that if the curve looks more like this, where it is above the detection threshold, your candidate for a brand new gene is a lot more likely to be because it is a brand new gene and not because it just mutated by accident.
Yeah, exactly.
Very cool. Okay, so
yeah, here we go.
Yeah, so this is where I got a little bit confused. I'm gonna be honest, because I don't have background here. So if you look at SPO13 using your math, you say it yourself, right? It cannot be eliminated, or I can't reject the. Oh god, the statistics language is confusing my brain. Basically, like there is an argument to be made that SPO13 is still a very good candidate for de novo gene status, but actually in the paper you point out that SPO13 isn't actually de novo, and I'm just gonna let you maybe go with this because I'm confused. I don't know what this means.
Yeah, so like I just said, so it's here all we've done is say we can't explain why you don't detect SPO13 using just by the sort of homology detection failure process that we've invoked. But it could be anything else. It could be de novo. It could be that it suddenly accelerated in its evolutionary rate outside of those species. And so uniquely in yeast, this is one of the many reasons that yeast are so great. They have a wonderful property that, as you've been saying, there's a sort of group of around Saccharomyces cerevisiae. It has all these closely related friends. And so, and in addition, some species change the order of their genes on the chromosome a lot. Like flies actually do this extremely rapidly. But yeast, or this little group of yeast around Cerevisiae, which are its friends, tend not to do that very much. And so they have this wonderful property called synteny, which conserves synteny, which is referring to the order of genes on the chromosome, which basically means I can look to the gene that's to the right of SPO13. I can look on Cerevisiae. I can look to the gene that's just to the left of SPO13. And I can say, okay, well, I can't find a homologue to SPO13 in Castelli, the species that you've drawn below. But I can find homologues to most genes, as we've seen, right? Because they blew that detectability threshold out of the water. And so because most genes are in the same order, I can take the gene on the right and the left, find those genes using the same algorithm, Control F, in the different yeast Castelli. And if those two are very close to each other and there's a little gap in the middle, I can say, okay, well, if assuming that everything is in the same order, if there is a homologue to SPO13, it should probably be in the orange space that you've colored there. And I can't see it statistically if I do the Control F against the whole genome for statistical reasons. Because like in short, I would have to correct for the fact statistically that I'm doing a Control F against like all of the 6,000 genes in the genome. But if I have a strong prior assumption that it's in that orange space based on the syntenic genes, I can just do one search, and I need a lower amount of statistical confidence, basically because I'm not having to correct for the fact that, like, if I do a test, if I flip a coin 6,000 times, I'm likelier to get a heads, even if it's very unlikely on an individual flip. And so this gives you increased statistical power. And so you can actually do this search. If you do the blast search of SPO13 against the entire Castelli genome, you don't find anything with high statistical confidence that looks like a homolog. But if I compare it just to the DNA sequence in between the two genes on either side, I actually do find something that looks very statistically like a homolog. So this is a way to sort of orthogonally test whether or not this gene is de novo, that is sort of only possible in yeast and a small number of other groups. And here we do find that it has such a homolog, which implies that it's not de novo, but it does seem to be accelerating in rate, or something weird has changed that rejects our null model in this species. So I sort of like this example because I'm indeed in the first part, like not immediately gatekeeping this gene as de novo, but this is sort of just a caveat emptor message that don't overinterpret the results of even this statistical test, because even if genes pass this initial screen, it still doesn't mean that you can run home and say that they're de novo.
I see. So instead of looking at SPO13 and controlling F for SPO13, you're looking at its neighbors because yeast preserve the order of stuff quite well. And I guess the implication is that yeast don't typically do things like insertions, or not. Maybe that's not fair.
Not as much. Yeah, not as much. They do have like all biology, it's complicated, but
Yeah,
it's
spectrum.
certainly not as much as a lot of other groups like fly. I'm working in flies now, and like flies just have an incredible amount of this, especially some flies.
Right. I mean, is it because of things like transposable elements, like just things flip in order, and or like what is the
I think
mechanism
so.
behind
I
it?
mean, yeah, in flies, I mean, in all, I think all, I don't want to say all, I think a lot of animals, which are much richer in transposable elements, do have higher rates of this. But I think it's still but it's not the case that all animals with high transposable element numbers in their genomes have very high rates of these sorts of rearrangements. It's probably a function of like DNA damage is required. Usually it happens when you have a break in the DNA and it gets repaired in some weird way. So rates of DNA damage affect it, and that might depend on like whatever chemical conditions are going on in your germline. So I think it's sort of still an open question why exactly these rates vary so much. But transposons are definitely an important part of it.
Gotcha. So there's an interesting question in chat, but just so that I fully understand the scope of the epistemology of this argument, this synteny argument, it's that it's not that SPO13 can't be de novo. It's just that it's really unlikely to be de novo because the chance that a yeast inserted something completely new there is pretty low because yeast don't like do that kind of thing. Is that fair?
Oh, I would say like it could still be de novo and older, but here what we find is that there's a something that looks like it's coding for a protein at the orange place in Castelli. So it's like has the same neighbors, and it looks very statistically similar to SPO13 if you only do the one comparison and don't have to statistically correct for looking in the whole genome. So the conclusion is that there is what I will call a cryptic homolog in Castelli. It's something that you couldn't recognize as a homolog if you were doing like 6,000 control Fs against the whole Castelli genome with SPO13. But when you can limit yourself to one and sort of take the reduced statistical burden, maybe that's a confusing concept, then I'm like, okay, now I can see it. And so it's probably
Gotcha.
not de novo.
So maybe it's like SPO13 and this other thing in S. Castelli. It's like they're like two siblings that look nothing like each other, but you know their parents are the same,
Yeah.
basically.
That's a really, you really have a talent for simplifying concepts that have had too many words used to explain them. Yes, that's exactly it. I have like a strong prior reason in the nature of their parents.
Fantastic.
Yes, exactly.
Fantastic. Or in this case, they're neighbors. They live,
Exactly,
you can
yeah.
conclude they're in the
There's
same
texture.
house because they have the same neighbors. Okay.
Exactly.
Okay.
Yeah, that's lovely. Yeah.
So we have a question from Eli. Oh, so the synteny test is amazing. Could you then look at the graph from S. Cass's perspective to see if R did indeed increase?
Yeah, I don't think we published that, but I think I have that somewhere. And yeah, it does. And I did a
Ooh.
lot of, it's on the paper, but I did a lot of work at like looking at the whole tree and trying to figure out exactly on what branch the rate increased and due to like mutations where in the protein. And yeah, your intuition is exactly right.
Sick. My audience is pretty smart.
I strongly agree. Yeah.
Yeah, you heard it here first, chat. Oh, hopefully not first. Hopefully this is not the first time you've been told you were smart. Maybe one of many. Okay, so now we have the gatekeeper for the status of de novo. So, Kara, I just want to do like a time check. Are you good for like a while, or should I like pick up the pace?
No, I'm good.
Okay.
Yeah, I'm not doing anything else today. I'm actually sick. Can you guys hear? I have like a cold today, so I'm at home on my couch. So yes, I'm
Doing a
happy to go on.
admirable job. Okay, so now we have your actual tests for the status of de novo. We just witnessed one candidate pass and one candidate fail, so we do have like it's stretched to say maybe robust because, as you said, things can leak through. You can have false positives, but now you have a test, which is better than not having any test, which is great. And it's easy in the math. Like you know, I can understand the math, and that's wonderful. So what we can conclude from here is not every unique sequence can be assumed to be unrelated to any other gene. Sometimes you just run into that opaque black space beyond the observable universe, and you just can't tell the difference between whether or not things are related or not. And so this is just like throwing it back to the giant virus problem is like maybe fast mutations explain why giant viruses have so many dang orphs. And I don't know anything about giant viruses that would lead me to believe that their mutation rate is higher. But maybe the fact that they're so big makes DNA repair problematic. I don't actually know, just hypothesizing.
Yeah, that's a good question. You mean compared to other viruses? Yeah, I don't know. That's a good question.
Because I know RNA viruses mutate like there's no tomorrow, but maybe there's something about having an enormous, like big chungus, you know, genome. Like maybe it's just hard to repair. I don't know. It's fun to think about though. Like at the border of biology, you get to do all this hypothesizing, and it's a lot more fun than learning, you know, the steps to mitosis. So I had a lot of fun thinking about that. And maybe we'll talk about that in the Q&A of like a word for the students out there. Biology will get more fun.
Yes, I love, like I said, I teach a freshman class on computational biology and statistics. So yes, I have another soapbox for that. I would love to talk about that.
All right, awesome. When we get at the Q&A then. Sick. Yeah. Okay, so this brings us to this question. Now that we have this test, how many de novo, this should be how many de novo gene candidates are there if I'm being super robust about the language? Because this isn't really correct, right? As we've seen, this is one gatekeeping test. There are many more that should be done. But how many genes pass the sniff? We'll call your test the sniff test. Is that fair?
That's good. Yeah. Yeah.
Yeah. So how many of these genes pass the sniff test in Saccharomyces cerevisiae? This part I'm not as confident of because I do admit I was kind of burned out. I was like rushing to finish this. So
everyone tells me that this paper is too long, and for 5 years I denied it, and now I'm like, yeah, it's definitely too long.
I don't think it's absolutely too long, like in terms of number of words, because I think the number of words is just about right. Just like my brain was tired when I got to the end of this. Okay,
that's a form of too long.
Yeah, that's true. So there were 25 candidates that passed this sniff test, right? And then there were 13 of those candidates. Oh wait, sorry. No, I'm no longer remembering the end of this paper. Yeah, wait, yeah.
I can't even remember the numbers, which is really embarrassing. Like it was so long ago that I'm like, what was the final count? I don't know. I'll just have to take your word for it.
But the idea is that you had a pool that passed the sniff test, and then you ruled some out due to other non-control F methods like synteny. Yes. And then 4 of those 25 had paralogs, which resulted from duplications within the same species. So I'm actually kind of confused as to how the paralogs did not get caught by the homology test.
Yeah. So one possibility, I actually can't remember for these, but there's sort of 2 classes of possibilities. One is sort of what you might call user error, which is often people like sort of only do exactly the test that you talked about, which is I start from some focal species like Cerevisiae, and I blast its proteins against all the proteins from some other species, but I do not do the test where I blast its proteins against its own proteins. And so you would just miss paralogs in that way. And so that's one possibility. The other possibility is that the paralogs, even within the same genome, that the paralogs have in fact diverged so much that they can't be sort of mutually recognized.
And so in that second case, would you still be able to find that with this like basic control F, or that's also like a black box?
With the blast method, not the synteny method.
Gotcha. Oh, okay. So now I'm understanding why synteny is so powerful in yeast. I get it now.
Yeah. And it's like, this is one of the things that will get much better as we sequence more and more species. Because like for some species, even if you sequence the species next door, your synteny still does not exist. Like in the fly I work in, that's true. But in some species, the more closely related species that you sequence, the more synteny that you begin to be able to see. So this has been a really big advantage of like the fact that biologists are now going out and sequencing every organism that they can get their hands on.
That's awesome. Okay, so then I do appreciate this little bit of whimsy, I think, at the end, where you then look at what these unique genes do using, what is it, gene ontology. So like you have this candidate list that passed the sniff test for de novo gene status, and then you had found that they, had like a vibe to them, right? I think you had mentioned
They
that
do, yeah.
they're like spore forming in meiosis. I don't know if you saw anything else besides spore forming. I think you had it in a supplement that I didn't read.
Can't remember if we talked about in Drosophila, we saw some enrichment for like relative to mosquitoes as outgroup for like CO2 sensing, which sort of implies, it could be which sort of implies that there you can't always tell which way the shift has gone, but there's sort of an interesting, tantalizing story there about how mosquitoes sense CO2 differently than fruit flies, and that we know that to be true because that's how they like find humans to eat. But I don't think we put that in the paper. But in yeast, that's the only thing.
Yeah, which is I don't know if this, yeah, because now I think we're in fancy woo territory of like this is just fun
Yeah,
to
yeah.
think about, right? There's
Yeah,
nothing
exactly.
robust about this particular statement here, but it does engage
Yeah.
the imagination of like I wonder what it is about the process of meiosis that and I guess to back up, these are not like well characterized genes, right? This is gene ontology based off of what they think these proteins do. Is that true?
Yeah, I'd have to look. I mean, this yeast in particular has the advantage. It's definitely the best studied eukaryote, and like yeast are really experimentally tractable. There's something called the yeast deletion collection where
Oh,
every
I
single gene in
remembered.
yeast has
Yeah.
been knocked out. Yeah, and there's been like at least basic phenotyping, like what does the cell look like? Does it die in various conditions? So we know something about like almost all yeast genes, and that's all the genes that start with SPO13, SPO16, whatever, have defects in sporulation, which is the process yeast have this very complicated thing where they can be both haploid and diploid. So you can like fuse two haploid cells to get a diploid, and then they will resporulate to form haploids. And this is how they sexually reproduce. Yeast can reproduce either sexually or asexually. So there's like all these SPO genes that have some defect in sporulation when you delete them. So there's something known about like basically every yeast gene, but you're right that like sadly for most of them we don't have very detailed molecular characterization.
I just had a wow, because I've been out of doing biology like professionally, I kind of had this feeling that yeast were going the way of the dinosaur experimentally. But you bring up such a good point of like, yeah, as a eukaryote, having a deletion library of everything, that's pretty powerful.
It's amazing. Yeah, it's really just a whole different when I moved from yeast to flies. I mean, I'm doing a little experimental work in flies, but I was never really doing experimental work in yeast, but just appreciating how much we know and what sorts of experiments you can do. It's a totally different ball game outside of yeast. So, but I think your perception's not wrong. Yeast are viewed as sort of like archaic and uninteresting, and it's very hard to get funding for them. And it's sort of a tragedy because you can just do so much in yeast.
Yeah, I just yeah, the deletion library seems like such a powerful idea. Very, very cool.
Yeah. Yeah.
So we're at the end of covering the paper, so I'll just summarize the paper in my words, and then we'll get to some fun chat. So de novo gene birth, a new gene popping out from the ether, kind of hard to prove, turns out, because of this epistemological problem of, well, is it really brand new or did it just mutate a lot? And there's not a really great way to distinguish between the two up until this very straightforward mathematical model that Cara has made. So sometimes having no relatives means your gene has been abandoned or lost by the fact that some proteins mutate too fast to be detected, right? There's a hard information cap that is represented by mutation rate. This isn't a matter of a lack of technological advancement, right? We're not being like, oh, we just need to look harder. I mean, actually, from our conversation, maybe you can look smarter, not just not with our basic control F
Harder, but
method.
not harder. Yeah.
Yeah, maybe there's a fancier control F that you can do that can detect this, but not the basic control F.
Yeah.
But a hard limit in information theory with regards to the basic control F. So at what point do two strings of text no longer look similar, right? There's that hypothesis argument to be made too in terms of the detection limit. So all gene candidates for de novo origin should be double checked, should pass the sniff test to see whether or not they could not as easily be described as having just been a natural consequence of mutations over time. So Cara is the gatekeeper of new genes, but a gatekeeper that I think deserves to be there and actually makes the story more interesting. That way, it's like these little treats, these little candies, right? And you might think you like the candy, but if you had to work harder for that candy, like we know that that's gonna make that candy taste that much better. That's a weird metaphor I just came up with, but I'm sticking with it.
I like that metaphor. I think de novo genes are pretty much like candy. They're
Yeah,
exotic and sweet. Yeah.
and treats and bad for you if you eat too many of them uncritically.
That's true.
All right. So here's a big philosophical question for you then. Could we ever prove a gene arose de novo?
Yeah, I think my current work, I think even before I would have said there are a very restricted set of conditions under which you could prove that. One thing that you would need to have is what actually BSC4 has. So one of the reasons it's sort of like the poster child for de novo gene birth is that, like, if you sort of imagine the molecular process by which a gene should be born de novo, you have some, as you said, like junk DNA that currently doesn't code for anything. And at some point, an ORF, exactly as you've said, should pop out of that. So you should go from having, so an ORF, and as you said, the ORF is a segment of DNA that has particular paperwork, certain like sequence characteristics that enable it to produce a protein. So you should be able to sort of line up a species that exists from before the protein evolved from that region of the DNA and the species where it currently exists. And you should be able to line up the pieces of DNA and say, okay, like it's clearly the same DNA, but I can pinpoint a couple of mutations that give it that correct paperwork. And so I'm sort of directly visualizing molecular events of the gene birth. So because DNA that's not currently coding for anything evolves so fast, that's sort of extremely hard. And that's why the fact that you can do that for BSC4 makes it such a poster child. And so if you had that, many people would say, like, there's reviews and stuff that refer to that as a gold standard for proof of de novo origination. I hedge a little bit there because actually the sort of vocal minority paper that you referenced for BSA4 and a couple of other examples, the line that he takes is not just that there might be unappreciated cryptic homologs, but he specifically gestures at a particular hypothesis in which genes are duplicated, genes that were previously sort of fully functional are duplicated to somewhere else and then die, or just sort of die in place because they're no longer useful. They sort of degrade very far. And because they're not useful anymore, there's no pressure from evolution to maintain their sequence. So their sequence sort of degrades, evolves neutrally. Evolution isn't trying to prevent the sequence from changing. And then once the sequence has like sort of degraded a significant amount away from the original sequence, like it looks statistically maybe like an unrelated gene, but maybe you can imagine there's some traces of functionality still sort of hidden in that sequence, even though it's very different from the original protein. Then you might imagine that that same region is sort of resurrected as a new gene. So it's sort of like fertilizer. I have like coherent, like an apple or something, which is a full apple. It's like a sort of, evolved fancy piece of matter. I put it into my garden, it composts, and then once it's sort of unrecognizable dirt, something new grows in its place. Is that a new thing? Or is it, as I'm sort of alluding to, is that only enabled by the fact that you had the apple or the functional gene there in the first place to sort of seed it? And so that is the part that really makes me hesitate about ever proving that a gene arose de novo, because you would have to sort of exclude the possibility that sequence that by all sort of statistical measures doesn't look like any protein coding gene was there, died, and sort of was resurrected in a very different form to form the new gene. And I'm especially concerned about this now because my current work is sort of suggesting that this is perhaps a much more common possibility than we have recognized. So I think this would be very hard. And one experiment that I proposed in grad school that my advisors were like, that's totally insane and you should not do that, but I hope someone one day will do, is to do a test where you insert a large sequence that you have made to be a random sequence, so you know that there was nothing there before, and insert it into the genome of like a yeast or an E. coli, and do the sort of lab evolution that you referenced earlier from the Lenzi lab. And if you saw something, a new protein emerge from that, you would know with 100% certainty that that's a de novo gene and that it's possible. But short of that, there may be special conditions under which you can do it, but I think it would be very hard.
Oh, that's a cool trick. I like that experiment. That's neat. It's not
Thank you.
It's not a complete argument, right? It just shows that it's possible, because
Shows that it's possible. Yeah.
the thing about you gatekeeping the status of de novo, like I can feel the anxiety, the like epistemological crashing out of like, is anything real? Can we ever do anything? Can we prove anything? If you can prove that it is at least possible, that keeps the conversation of de novo gene birth on the table, because you're not anti de novo gene birth. You're just, we got to be careful about it.
Right. And in fact, like it must have happened at least once in the history of life, right? Like the extreme case, just it's probably not this, but the extreme case is that there was one gene born at the beginning of life and everything else is sort of duplications, exon shuffling, derivatives of that. But it must have happened at least once.
I did get that comment in my YouTube
right.
video. Like it had to have happened at least once. Like that's true.
Yeah. Yeah, exactly.
But isn't that such a funny realization of like, we know so much about biology now that we didn't before. Like every cell in C. elegans has had its like lineage traced. Like you get like a worm, a C. elegans worm, and you can trace, the fate of every one of those cells. Is like one of the craziest things that I've ever heard of. Like as it's developing, that cell is going to be this neuron in like 2 days. That's an insane amount of fine resolution detail we have about life today. But in terms of like, how many times did a new gene pop up? I don't know, at least once.
Yeah, right. That's more uncertainty. I know, it's sort of incredible, isn't it?
Yeah, that's awesome. Okay. All right, so we're in the Q&A section now. If you want to just hang out for 15-20 minutes, we can talk about whatever chat wants to talk about. We have a very special guest. I would take advantage of the fact, if you have any deep thought-provoking questions about the study of genetic novelty, or what is it like being a professional scientist, now's your freaking chance. But as chat is coming up with their questions, I'm wondering if there's any like wise words you have for any of the students in chat, because we have a mix of, I want to shout you all out. Like I'm so blessed to have such a varied audience of like biology students, but also a lot of people who have not been formally trained in biology. And like, what are you even doing here, man? This is like a high level, graduate level, like pushing the boundary of what we know. Like, unlike a science journalist that would just give you the top line, we walked through all of Cara's math and her data. And if
I was really impressed with your level of detail. Yeah.
Thank you. And I am proud of you for sitting through all that, because it's not easy. And if you didn't get everything, that's to be expected, because this is hard. And I needed to go to school for 1,000 years to understand this. So, yeah. Oh, we have a music major in chat. That's crazy. It's just awesome. Insane. The thing is this new era of like science communication is assuming that the audience is smart, which I do every day. I'm assuming, and a building designer too. Just crazy. Cool. Very cool. So I made the last scientists I invited on do this. What would you? Well, I was also a student for a very long time, a very good student. What would you give me? Test score. Grade my journal club.
I think what is you want a letter grade? You want a number out of 100?
Give me a number. That's more precise. I would say.
I think I'd give you like a 95. You can never give anyone 100. That seems like bad pedagogy. There's always somewhere we can improve. But no, that was like incredibly accurate and well paced, and is much more detailed than I give when I give talks on this. And yeah, it was great.
Thank you so much for the super chat, Hair Day. The algorithm has led me to many places. I have no doubt that it could lead non-scientists here. Yeah, that's what I'm so appreciative, and I'm so happy to share this, because I imagine, Cara, when you usually give talks, it's probably not to people who aren't biologists, right?
No, yeah, very rarely. Yeah, and I would like to do that kind of stuff, but you've probably heard people complain about this. It's sort of very hard, even for like scientists who want to engage the public, to find the right venue for it. So I'm really appreciative, and yeah, it's a very different experience. It's been fun. Yeah, some of the great questions coming from the chat too throughout all of it. While people are coming with questions, can I ask you how you kind of tumbled into biology?
and what your
Yeah.
journey here has been like.
Yeah, I love to talk about this because it's a very non-canonical path. I
Mm.
started, like I alluded to briefly, I started life really thinking I wanted to study philosophy of mind. I wanted to understand like the nature of consciousness and the human mind. And so I was, like, okay, I'm gonna do philosophy, but I should probably take a neuroscience class because that's relevant. And then I realized the brain was made of neurons. So I was like, I probably understand something about neurons, which are cells. So I took some cell biology and I was like, cells are basically run by DNA. So I took some molecular biology, and then I was like, okay, well, it's run by the DNA. What dictates how the DNA changes? And that's what eventually led me to evolutionary genomics. And then I was like, this, as you said something really beautiful earlier about like evolution being the story, the causal story of all of biology. And that's like very deeply my view. I just think that there's a quote from Dobzhansky, who's a famous evolutionary biologist, that nothing in biology makes sense except in the light of evolution, which is the process that generated it. And so, like, my sort of long, very long term, we're talking like, 10, 20, 30-year goal is to return to studying the brain. But I feel very strongly that the way to understand any biological system is to think about evolution and the way that evolution acts on the DNA.
That's awesome. We actually have overlap there where I mean not quite a lot. Where we intersect is probably an interest in the brain. So I went to graduate school for neurobiology, and
Cool.
there is something very compelling about the organic mush that sits on top of our shoulders.
Yeah.
On the consciousness, I did not dare to do that. No, 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 no.
And I actually applied to cognitive neuroscience PhD programs in addition to all my like normal biology ones. And I remember walking into an interview at Duke. Like they let me in actually, but I walked into several interviews at Duke and I was like, I want to study consciousness. And someone literally said to me, like, you can't talk about that in polite society. And I was like, okay, the field's not ready. Bye. And it's
That's
been really
so
heartening
funny.
now, actually. I sort of moonlight at a couple of consciousness science conferences. So it's like sort of coming back. But yeah, it's like still sort of a dirty word.
Yeah, I actually didn't know that from a cultural perspective. It's just my personal take on the consciousness problem is like I don't know enough math to be able to model or contribute. Like my thing was more like in terms of like strengths and weaknesses, like I really like you can tell like through my communications that I really personify things, which is
Yeah, yeah.
like a weakness for certain kinds of thinking, but I think a strength in others in terms of narrative forming and like sniffing out where to go next, thinking about the narrative of the cell. Yeah, I was more interested in like how certain proteins in the brain can affect like someone's lived experience through cognitive changes, which is like different than consciousness, even though cognition and consciousness, they are distinct ideas. So I just think it's interesting, kind of the overlap of this like not shying away from some difficult problems.
Yeah, but it sounds like a core interest of mine is present in your interest too, which is like mapping the material onto something about the subjective quality of your experience. Is that right?
Yeah, for sure. Like I always
Yeah.
felt it was like compelling to I had a conversation with a philosopher VTuber about the nature of free will, which is very much tied into this idea of consciousness. This is this is kind of an out of pocket for a Q&A, but as like people in chat ask questions and stuff, yeah, why not? Like it's interesting to see people's perspectives, to kind of, I'm not going to give any more context, but like what is your take on the whole free will thing?
I guess I feel like I think about free will. I guess I feel like it's sort of like a language game or a language misunderstanding, like we were saying earlier. I guess I feel like depending on, assuming we're all sort of like good, quote unquote, good materialists, like meaning that we don't think there's any causal stuff outside of like, physical particles, which I'm actually not sort of committing to that view. I think consciousness is sort of like a wacky enough field that personally I think David Chalmers' view that it's like something more akin to another fundamental force in physics is probably right. But I guess I think most people are sort of good materialists in science. So if you're a good materialist, I think I just don't know what you can mean by free will if not some sort of like causal structure arising from matter moving around. And that's a sort of highly diminished view of free will to me. So I think people just, I don't know, I think it's sort of like confusion about language. That's not a very good answer.
No, it's a great answer because that's similar to what I said. Like 2 for 2 in terms of biologists on this who have appeared on this channel. That like free will is like, to me, it's not okay. Do you think free will is an interesting question or not worth thinking about?
I think it depends on your like presuppositions about the sort of causal nature of the brain. I think if you're a good materialist, I don't think it's that interesting. And then if you're not a good materialist, I think depending on the specifics of your review, it could be interesting. Like if you believe there's some like equivalent of the soul, which is like, where a sort of non-material force interacts with the brain, then maybe there's room for a classical conception of free will. But I think if you're a good materialist, it's sort of very uninteresting.
Yeah, I would put myself in that category of like, I think I just said like, I think it's important that people believe that there is free will, but whether or not free will exists or not is not exactly
Yeah.
interesting to me.
Yeah, I strongly agree with you there. I'm a sort of utilitarian morally, and from that perspective, I think it's essential that we believe in free will.
Which is it's interesting having conversations with philosophers because a lot of good philosophy questions are rooted in some aspect of the lived reality of biological existence, and I find myself just answering this is not an interesting question for a biologist to the majority of their questions. It's just really shocking in some regards because so many big questions out there that people love thinking about when you look at it through. I'm not saying all biologists believe this, but if you take the biologist lens, the materialist lens, as you've said, a lot of those questions just kind of lose their luster. I'd rather think about whether or not new
Yeah.
genes are a thing.
Yeah, well, I would too, obviously. Yeah. But I think that's a really essential skill in science, maybe to get at the sort of soapbox of science pedagogy. Like, I think it's almost as important as a scientist, I don't know if you agree, to figure out what questions are not interesting and rule those out, even if they're sort of big, flashy questions. That's like a very important aspect of scientific taste to me.
Yeah, there's a scientist that I worked with at the university that I last taught in who had said that one of the most core skills that a mentor can help train in their student isn't like a robust knowledge of the cell cycle or anything like that, but rather the ability to identify good questions. Yeah, as you said, not flashy questions, but like meaningful questions to answer. And that's always stuck with me because like, if I were to grade myself on my own ability as a scientist, and this is not talking down to myself, I like on paper, like I objectively was a bad scientist. It's not like a pity party. Like the skills required to pull off.
Curious how you like do that objectively, but okay, that's fine. There's so many types of scientists, but
I think that like if I'm defining a scientist who as someone who does benchtop work and publishes papers like that, I was not great at that. Which,
I don't know if that's everything that makes one a good scientist.
no,
But
no,
in the
no.
modern day, especially, I can see why practically the forces collude to make that seem like the definition of a good scientist.
Yeah, if you buy that the evidence of being a good scientist is frequent publication in high impact journals, then I think I'm comfortable saying I was not that. But what I do think I'm good at is identifying questions that people will find interesting. Maybe not what like a funding institute wants to fund, maybe not something that revolutionizes all of cancer treatment. Like I think I've picked my lane and I'm very happy and comfortable in that kind of lane. But that skill is something that you don't pick up just looking at lecture slides. It's like it's active cultivation of a philosophy, a praxis, something.
Totally. Yeah.
Okay, we actually do have some questions in the chat that we should get to now. This work was very computational/statistical. Have you done or are you currently doing any wet lab experiments too? That's an interesting question. Do you do wet lab work?
What a great question. I'm so proud to give a positive answer to this. Yes, in fact, I will give you like a totally mind-blowing anecdote that I never could have predicted even a year ago. I'm like I mentioned, I'm now working in Drosophila, fruit flies, and I'm now working on a problem that has ultimately led me to the process of exon shuffling. And I'm working on regions of the genome that seem to do something like exon shuffling extremely quickly, such that even comparing different species, like too many mutations have happened and you can't really see what's going on. So I was like, okay, I need more individuals of this one species. And the species just happens to be native to New Jersey, where I'm currently living.
Wow.
So for the last year, I have been going out into my backyard, catching flies from the wild, bringing them into the lab, establishing lines, and sequencing their DNA and RNA. So change is possible. I was previously a totally computational biologist, and now I'm like mucking around and rotting fruit in my backyard.
That's so sick. That's so sick. That's awesome. Um, Let's see what
Unbelievable.
else. Other questions we've got. We've got some questions about organ transplantation. I feel like that's a little outside of the
Hmm.
scope of our expertise because that's more of a biotech kind of problem.
Yeah, I'm not a biotech person.
Yeah, that would be, I think, the limitation. Of, I mean, we could shoot the crap and guess, but yeah, I don't have any reasonable. I don't have
Your
a
call.
well
Your
informed
party.
thing to respond to that with. Let's see. Uh, let's see. Any other questions here? Oh, we have some philosophy in chat too, which is fun. Yeah, making your own lines from the wild is also really cool. Very unexpected. I remember working across the lab, across the hall from a Drosophila lab, and every once in a while, I'd find one of these suckers.
Yes.
So funny.
Yes, they're quite quick and eager to escape.
How
It's funny, the
how
ones you catch in the wild are like way faster. Like I was also doing a little bit of work with sort of like well domesticated lab Drosophila that have been in the lab for, 60 plus years. And they've definitely lost some of their reflexes. I don't know if it's like a genetic change or what, probably to some extent a genetic change. They're really slow, and the lines I catch from the wild, like, they escape so much more often when I'm changing their food, so they're in my lab flying around quite a lot.
so like hybrid vigor or something?
I
Uh,
don't know, man. I mean, I think it's probably just that, like, after you maintain lines in the wild in from the wild in the lab for long enough, they lose the selection pressure to do all their natural, like, escape behavior because there's no use for it in the lab. That's something that you see in the Lenski lines, interestingly, right? Which is that the things that the bacteria don't need, they very quickly sort of delete from their genome because it, like, makes them, they don't need to, like, spend the energy producing it. So I think flies cultivated in the lab for long enough have just, like, ditched a bunch of behaviors that they don't need anymore.
that's true. They're not
But I have
exact.
no data to support that.
They're kind of being given a very chill existence in a lab and no longer actually need
Yeah,
those.
right?
Yeah, it makes
It's a
theoretical
good light.
sense. Yeah, that
Yeah.
does. Okay, I don't know how much you can reveal about your current research, but the idea of there being hotspots for genetic shuffling in these Drosophila, do you think that's mediated like sequence dependent, like a DNA side or like a protein side kind of deal?
It's a really good question. I have a preprint on this if anyone's interested. Preprints are
Ooh,
all open access, so that's great. It's called Exon Shuffling Hotspots in an Animal Genome. So yes, I'm very happy to talk about it. It's sort of hot off the press. I think part of it is part of it is exactly what you alluded to earlier, which was very intuitive of you. Part of it is definitely transposon mediated. So like having all these transposons in the genome that are very similar allows the cell to repair DNA breaks when they happen, sort of thinking it's using the same sequence, but it's actually like a copy of the same sequence at a different place in the genome. And that's how one of your listeners asked earlier, like, why do you get recombination not just between two alleles, but between different places of the genome? That's sort of how I think about it, is that the cell thinks it's doing allelic recombination, but like if the sequence that is mediating the allelic recombination is sort of repeated many times throughout the genome, it can get confused. So definitely part of it is transposon-mediated, so-called ectopic recombination in that way. But I think there's another factor, which is like you have to have the DNA break for this to happen in the first place. So again, it's like almost it's not this part is not in the preprint, but my intuition is that these flies just have a really high rate of DNA damage for reasons I don't totally understand, and that that's part of it.
and the DNA damage is like localized in certain hotspots, or
That's a really good question. I'm not sure. I think that could be it. Like we know, not in this species, which is it's not even Drosophila melanogaster. It's a very obscure species of fly, but we know in general, right, that DNA damage is likelier to happen on regions of DNA that are not so what are so-called protected. They're not like wrapped up in protein coatings like histones. They're sort of
oh
accessible to
god.
be damaged and then repaired. Yeah. So right. So we're really getting into like sort of nitty-gritty molecular
uh,
biology. I think it's something about like the accessibility of the regions, and maybe something about like in 3D space, like maybe you get damage, but you can't necessarily repair that damage using one of these other copies unless you're close in 3D space to an appropriate other copy of the transposon. So my current thinking is that it's going to be very complicated, but I would love it if it turned out to be simple. And I'm sort of like very much in the thick of this right now.
Yeah, so Wacky Lemon says the physical chemistry of a chromatin. That's why I like cringed. I was like, this
Yeah.
seems difficult. Because I was
Yeah.
like,
Yeah.
if
It's really hard.
if there was like DNA damage hotspots and it was sequence based, well, we could just control F. That seems pretty straightforward. But yeah,
Yeah,
yeah, having to
yeah,
think of how is the DNA wrapping around the chromatin, which parts are not chromatin exposed, and I'm getting all these flashbacks to graduate school of ChIP-seq and all that stuff, which I don't remember
yeah,
how
yeah,
those work anymore.
exactly.
But oh
Yeah, man, I'm thinking about having to do a lot of this. I did, yeah, I spent a long time trying to exactly as you say, like control F and find some clean sequence-based readout of where these hotspots are. And you get sort of like a weak signal, but it clearly doesn't fully explain it. So that's sort of disappointing.
yeah, man, biology's complicated.
Yeah. Yeah. That's what makes it sort of beautiful, right? Like these sort of dirty, nitty-gritty molecular details cascade up and give you these sort of incredible evolutionary phenomena. I know it's painful, but like I do sort of maybe it's like a massive it's like a Stockholm syndrome thing, but I've sort of convinced myself that that's part of the beautiful part.
I like this question about taste a lot because it helps, I think, students develop their own sense of taste if they're not entirely sure what to like, what path to go down. Because again,
Yeah.
I've talked with my psychologist friends who are with on V Theorem with me, and we kind of have this view of like, you know, when we as biologists talk about biology, we're like, oh, biology is so messy. It's just it's not neat. But then when you talk to psychologists and you're dealing with the human mind, it's like, oh, okay, we're actually very tidy with regards to what we can and cannot control. Because when you ask a psychologist, like they can't have control over like their experiments cannot have the same level. Like a biologist can freaking take an optical laser and immobilize a protein and tug on like a portion of a protein to measure the newtons like in between the bonds of a protein. Like that's an insane amount of control. Like you can
Yeah.
take your little molecular CRISPR tweezers and edit a DNA genome like very, very precisely. And you can do it with RNA too now with ADAR, right? You have all this precise
Yeah.
molecular control over our system. And I find that like trying to make order out of the mess that is biology is why I find molecular biology to be very satisfying. Because sometimes the path
Yeah.
to an experiment is really that straightforward. I told this story a couple times on stream, so I'll indulge myself again and tell it again. The reason why I chose molecular biology over psychology happened at a very specific moment when I was listening to a lecture by my advisor, who is like a kingpin in kinesin, like the motor protein kinesin, walks on two legs
Cool.
across the microtubules. And he was like, yeah, we needed to know like how this interaction between kinesin and the microtubules happened, so we just mutated everything in the feet. And I was like, oh, okay, I could do that. Like conceptually, like that makes sense to me. Like there's a surface area
Yeah.
contact between the protein and the microtubule. You just mutate that, see which amino acids are important, which one aren't, and from there emerges like a beautifully put together inquiry and story that pops out of that. Whereas I feel like psychology, it's not dunking on psychology. It's a matter of taste, right? Psychology is about making order of the messiness that is people's behavior, and that to me, there's this insecurity of like, oh my god, but how do we know anything is real?
Yeah. Oh, I totally agree with that. Yeah. I think I spent a long time in my personal trajectory sort of orbiting that point of tension. Like the subject matter of psychology was more interesting to me, but like you, I sort of shared that queasiness and I guess ultimately couldn't get over it.
Yeah, queasiness is I think yeah, because yeah, I was deciding between psychology and molecular biology as well, and yeah, pivoting to biology because it felt a little more secure, like into like what is it, the term I've been saying all stream, not pedagogy, damn, epistemologically secure, right? It felt like the line
Yeah,
between
yeah.
point A and point B made a little more sense. Not to say that psychology isn't robust, it's just, you know, humans are messy.
I'll say it. I'll say it. I'll go on the record.
Damn.
It's robust in its way, but I just, I have very little certainty that it sort of maps onto a physical reality that will endure long enough to lend itself usefully to a wide range of other models. Like as you say, every model is wrong, but some are useful. But if the domain of its application is sort of sufficiently restricted, it's less useful. I feel like it's just like a bunch of ad hoc models. Psychologists in your audience can fight me, and this is not Pai's view. He's not endorsing this
I
view.
am not endorsing this view at all.
Call me.
All right, you know where to send hate mail. No, don't actually.
Yes. I'm fine with that.
Yeah. So Dragon says, you know what I love? Behavioral ecology and ethology, just slightly less messier than human psych. Behavior. I mean, like from a neurobiology perspective, behavior for me was, because I view myself compared to a psychologist as like a control freak, right? Behavior to me was always like, how long does it take for a mouse to escape a maze? That's behavior. Does the mouse remember where the cheese is? That's a behavior. But anything more complex than like these very basic patterns, I felt was too scary.
Yeah. Yeah, I think I know just enough about it to like feel that I like it and have not looked into it anymore to dispel that illusion, which is a dangerous place. But the ethology part of that really resonates with me. I guess I'm only sort of articulating this now as I realize it, but I think one thing I really like what appeals to my scientific taste, is when you have a sort of like macro level phenomenon that you find sort of interesting in itself, like evolution or behavior. And then you find a model that maps on to a structure in physical reality that has a lot of explanatory power. For me, like the DNA and changes in the DNA, I find that really satisfying. And ethology to me feels like, as I think about it from 1,000 feet away, feels like the analog of that for behavior in the brain. Because like, all these like original ethologists had these wonderful sort of hierarchical models of behavior, right? Where you have like the nesting behavior, and that breaks down into like a series of stereotyped actions, which further break down to a series of stereotyped sort of like neural circuit or spinal cord circuit pathways. And so I feel like if I were going to do neuroscience, I would do ethology, because that feels like the place where the phenomenon like touches the anatomy in a way that is really satisfying.
This is an experiment that I promised I would never cover as a video because it's a little too controversial, right? Mapping like very precise, because people get, okay, if you're in my streaming audience, I assume you will take the knowledge that I give you responsibly, right? But I can't guarantee that with the algorithm, right? But in terms of ethology, I remember learning about this optogenetics experiment where you can optogenetically modify like a specific neuron in a mouse brain, and when you shine a light onto that neuron, it'll just make the mouse turn left. That's crazy.
That's beautiful.
Like
Yeah, that's beautiful. I love that.
they've identified the turn left neuron. That's how crazy this kind of science can get. I love it.
That's wild. Yeah, brains are so weird. That's wild.
It's like on several levels where like, yeah, your personality, your thoughts, feelings, and emotions, that's all pretty complicated. But sometimes, very rarely, you get the turn left neuron, and that's just sick.
Yeah, we live for those moments, right, where it looks simple.
Yeah. Hoping and praying that it's simple. And sometimes
Yeah.
sometimes it is, and that's dope.
Yeah.
Oh, yeah. But yeah, you can imagine if I did a video on that, people would be, oh, mind control. It's like, well, I mean, it's not even that like crazy, right? Because we've known, through like Phineas Gage, and all the medicine that's arose around, traumatic brain injury, that yeah, there are certain parts of the brain that control certain things, and if you damage the brain in certain ways, some weird stuff is going to happen. So there is that like physical component that I do find very fascinating, of like, this complicated phenomenon that we call consciousness. Just a chunk of it can be explained just by neurons being neurons, and I think that's cool.
Yeah, I think that's incredibly cool. I mean, I know that's sort of like unromantic to some people. There's this phrase from I think Keats that's like, you're unweaving the rainbow. But to me, there's like really nothing more beautiful than to unweave the rainbow. I just like I really live for that as a scientist slash former philosopher, I think.
Yeah, because this is again like the philosophy of and taste, right? I keep on repeating this term taste, because I'm very diplomatic. I'll say, but it is a matter of taste, and especially because this is a teaser for the abiogenesis video, is that there's a sense that if at the center of the abiogenesis question, if it isn't divine, then it's kind of boring. But I think it's the exact opposite, where this is it can be controversial. It's okay if you think that the center of the abiogenesis problem is divine. I have no issue with you. It's just that I think that if you acknowledge that the center of the abiogenesis problem, I'll test this out. You can tell me if this is too controversial, Chad, and I'll leave it out of the video. This is special for you, Cara. You can be a testing ground for this idea, because I trust that the streaming audience isn't gonna like take this too harshly, but I view biology like a candy, like an infinitely layered candy. And at the center of the candy is abiogenesis. Everything that we do, everything that comes out of biology is all retracing the narrative of evolution from the frame that life popped in, right? And even before that, right? Chemical soup, prebiotic soup, vents, warm little ponds, comets, whatever. The story of biology is this multi-layered candy, and at the center is abiogenesis. When you've worked through all the layers, vanilla, chocolate, the ERK pathway, mitochondria is the powerhouse of the cell, right? Each one of these concepts is its own additional layer, and at the center of it all is, okay, so how did it all begin? And I think if there's something I think that is compelling about not knowing if there even is a center. Maybe the center is hollow. Maybe, as you pointed out in your paper, there are just some things that we cannot know because of information theory, right? And so I have this idea where there is a center to the candy. It's either going to be the most delicious flavor anybody's ever imagined in their entire life, like it's going to be so cool what we learned when we get there, or it's going to be hollow and disappointing on the inside. Because we can't know, because there's just a limit to our methods of study. And I find that exciting. I don't find that depressing in the slightest. I find that the idea that at the center of all this beautiful biology is just nothing, or it's going to be sick. I don't know. Maybe I'm a gambler. To me, that is very compelling. And if you want to believe that center is the divine, that's, I bet the divine tastes great, but it's just, I would rather gamble on it, right? I would rather gamble on it.
Yeah. Yeah, I think I'm similar. It's never really, I guess I've always sort of assumed that we can't know. I mean, I'm not an expert in abiogenesis, but I've always sort of assumed that the answer is going to be the latter that you gestured at, which is just that it's sort of eroded by time. And yeah, I'm pretty okay with that. There's something beautiful about that too. It's some detail of the chemistry of the early Earth, or wherever life originated in our solar system, that was so idiosyncratic as to be gone. And that's sort of beautiful too.
Yeah, I think you can take it in a couple of different ways. Hairday says, I think it's going to start a flame war no matter what position you take. The thing is if I'm doing an abiogenesis video, there's going to be a flame war. It already happens on my videos that have nothing to do with abiogenesis. I get crap.
Is it really?
I get I get a decent amount of crap on random videos about evolution is a lie. I'm like, oh my god. Oh my god. Yeah. I didn't even talk about, I mean, if you talk about biology, you are talking about evolution, right? As we said, evolution is the narrative through line, that it is the beating heart, the soul of biology. But I didn't even make explicit reference to anything of evolution. I still get comments that really don't like this idea, and I'm like, oh bro. So I'm doing abiogenesis. I'm gonna get crap for it. I can already tell. It's just a matter of how do I mend the gap? Both perspectives, right? The flavor of the divine on the center, or the chance that it could be the most delicious flavor in the universe, or an empty nothingness. I think they're both valid, and there's a through line of taste that I keep on repeating, because I think belief, this is way more philosophical than I thought I was going to get into with a guest on, especially, but a belief is composite. It's evidence. Yeah, you see evidence in your world, you incorporate that into your mind model, but there's also a component of aesthetic preference, right? I just like the idea that at the center of the candy could be nothing. That'd be kind of crazy.
Yeah, I'm really glad you keep referring to taste, honestly, because I mean, I guess harkening back to your original thoughts about what professional scientists would say to students, and how biology is much more fun to practice than to learn as a student. Yeah, it just sort of makes me very sad when the creative aesthetic taste-based element of especially biology is not accurately transmitted to students. It's such an important part of being a scientist, to develop taste, not only to be successful, but to allow your own career to be satisfying to you, because there's a hell of a lot you could work on in biology, and you have to find some way to guide yourself. So I think it's very important, and I'm glad that you are doing your listeners the service of emphasizing that so much. I wish I had that more as a student.
Yeah, it's a little uncommon, especially because I think, as you said, taste is a motivating, it's a part of your scientific personality, right? If you're a working scientist, dude, you're smart. We get it. You can do the mental operations to put, whatever together. But what determines part, are you a great scientist? Part of that is your taste. And do you feel great about being a scientist is all about a matter of taste too. And I think I don't refer to what I do on the channel like teaching, because I feel like teaching is very like handholdy, like very like teaching is what I did for my previous profession, where I would have a PowerPoint, I would give you a quiz, and I would grade your quiz. Like that to me, that's teaching. This is more like I am trying to like help encourage a scientific praxis, which is not like you don't have to agree with me on everything, but like like thinking about the ways in which you disagree with me, that's important, because I'm not going to be right every time. My thinking is definitely not going to be right every time. And like, if you disagree with my sense of taste, that's also good, because by telling you my taste, if you don't like that taste, you've just identified something about yourself. And I think that's important. And I think that's cool.
Yeah. Right. And I mean, I think taking a lighter touch in pedagogy serves students better in terms of becoming a practicing scientist, because like there is this sort of weird paradox. This really like tangled me up several times in my undergraduate career, where I had several sort of existential crises, where I went to my philosophy advisor, my former philosophy advisor, and was like, I think I need to come back to philosophy because I don't want to do molecular biology anymore. And it was always moments where there was some appeal to authority to understand something. I remember a moment where we were looking at photo 52, which was this X-ray diffraction photo of the structure of DNA that Crick, a physicist, immediately looked at and was like, DNA is a double helix. But to interpret the actual photo as like a physical structure, a crystallographic structure, you need Fourier analysis, which is a very complicated domain of math. But in this biology class, of course, like for sophomores, they're not going to teach you Fourier analysis, but they want to give you some idea. And so they sort of inartfully just appealed to authority. They were like, look at this photo. This is obviously a double helix. And everyone else sort of like nodded along. And I was like, I can't do this. I have to be able to understand this. I must be stupid for not understanding this. I sort of could not accept appeals to authority in a science class. And I think you don't serve your students well when you do that, because once you become a grown-up scientist, you can't, like, you are a bad scientist if you accept appeals to authority. You have to learn how to reason through it for yourself. So yeah, I think the lighter, I like how you put it, but can't recapitulate it. Cultivating a praxis, that's like very valuable for cultivating effective scientists.
On the photo 52 thing, in graduate school, we did have to it was on our final it was on our final exam. I remember studying for that. I remember studying for that final exam where they showed us photo 52. It was like, map out why this is a double helix. And I remember the night before, as I was cramming for this final, I was like, if I get every other question on this exam right, I'm just gonna leave that part blank because I never understood how that worked.
It's really hard. It's really hard. They tried to make me and my cohort mates in biophysics take like a hardcore crystallography class where we did the Fourier transforms by hand, and we sort of like mass defected and went and took an evolutionary dynamics class instead. It's really hard stuff.
It's really difficult. It's cool that we were taught how to do it. I have no idea how to do it. Okay, with that being said, I do think it is time for us to call it a stream. Do you have any last parting words of wisdom before we let you go today?
I guess just because I didn't get to stand on this particular soapbox in terms of the math in the model, we characterize it as simple, and like from some perspective it is, but if anyone in your audience or residually you like has some quite queasiness about it, I guess I would just say that what one of your viewers said is absolutely right, that the most important thing for learning something is believing that you can learn it. And so I hope that inflicting this aspect of the paper on you was pedagogical and inspiring rather than punitive. And I really appreciate you working through it in as much grace and detail as you did.
Thank you so much. Thank you so much. And thank you for the high 95 mark too. I'm going to cherish that as someone no longer in school. All right. Thank you everybody for coming to hang out with me today. Say goodbye to Cara today. Thank you, Cara, for being here today. I think I've said your name probably 3, 4, or 5 different ways through the stream. I do apologize for that.
I think I heard it right. And my PhD advisor actually still calls me Cara, so you're doing better than him.
Okay, great. All right, thank you so much. All right, bye everybody. Say bye.
Bye guys. Thanks everyone.